When you think about carbohydrates, I bet your mind probably jumps straight to energy.
Yeah, pasta, bread, sugar in your coffee.
Exactly. We're sort of trained to see them as just fuel, right?
Yeah.
Calories you count, burn, store.
Mm-hmm.
But if we actually, peel back that diet talk and look at the source material, the chemistry itself,
Yeah.
it's maybe one of the most structurally subtle, mathematically complex, functionally diverse areas in all of biochemistry. It's kind of amazing.
It really is. It's an area where tiny details, millimeters if you like, at the molecular level, they matter hugely. Historically, people called them hydrates of carbon.
Right, the whole CnH2O formula.
Exactly, because often the empirical formula looked like that, but honestly, that definition is chemically pretty insufficient.
It doesn't quite cut it.
No, it's a bit of a relic, really. It doesn't hold up when you look closely.
So if that old definition is inadequate, what's the precise modern chemical rule we should be using?
Well, the sources are really clear on this. Carbohydrates are rigorously defined by their functional groups. They are polyhydroxyaldehydes or polyhydroxyketones or compounds that yield those specific structures when you hydrolyze them, break them down with water.
So the key bits are the multiple hydroxyl groups, the OHs, plus either an aldehyde or a ketone group.
That's it. That combination is the defining feature.
And we really have to use that functional group definition because the old formula just fails for some really crucial molecules, doesn't it?
Precisely. Take D2-deoxyribose, the backbone of DNA.
Can't get much more essential than that.
Right. It's absolutely a carbohydrate derivative central to life, but it doesn't quite fit the CH&H2O formula perfectly because it's missing an oxygen atom. That's why we have to rely on the presence of those critical functional groups.
Okay, so our mission in this deep dive then is to move way past the grocery list view and really understand the secret life of sugar. We want to explore how these incredibly tiny differences in 3D structure, what chemists call stereoisomerism, how they dictate absolutely massive biological outcomes.
Yeah, it's fascinating.
How does just twisting one single bond determine if a molecule fuels your run or builds the shell of a crab or even determines your blood type? It seems incredible.
It's the ultimate lesson in structural humility, you could say. We're showing that life's efficiency relies on absolute specificity. We'll show you why just one misplaced hydroxyl group can make an energy source completely unrecognizable.
And useless to our bodies.
Functionally useless, exactly.
Right. So to really get a grip on this complexity, we need to start with the basics, the scale of these things.
Yeah.
How we categorize them, primarily by size, how many basic units make them up.
We start at the bottom, the foundation, monosaccharides.
Okay.
These are the simplest sugars, the basic building blocks. You can't break them down further into smaller sugar units through hydrolysis.
And these often fit the old formula.
They often do have that generalized H2O matter formula, and importantly, they have all those classic physical properties we think of when we hear sugar.
Meaning, these are the sweet ones.
They're sweet, yes. They're crystalline solids at room temperature, and they're highly water-soluble. Think of glucose. Small, neat, dissolves easily in your bloodstream. That's a typical monosaccharide.
Okay, so moving up the size chart, what's the next category?
That takes us to the oligosaccharides.
Oligo, meaning few.
Exactly. These are molecules that, when you hydrolyze them, they release a modest number of monosaccharide units, somewhere between 2 and 10, usually.
2 down, okay.
Now, while that range is technically correct, the most common and biochemically relevant ones, the ones we'll focus on, are the disaccharides.
Dis meaning 2.
Right. 2 monosaccharide units linked together, like lactose in milk, or maltose from starch breakdown, or sucrose, table sugar.
Got it. And then from the few, we jump straight to the giants, the polysaccharides, or glycans, you call them.
Yes, glycans is another term, polysaccharides. These are the real structural titans of the carbohydrate world.
The polymers.
Huge, high molecular weight polymers, sometimes built from thousands, even up to 1 million monosaccharide units all linked together. Wow.
And structurally, functionally, they're really the opposite of simple sugars.
How so?
Well, they're generally tasteless. That's why we often call them non-sugars. Think about starch or cellulose. And instead of dissolving clearly in water, like glucose does, they typically interact with water to form sticky, viscous colloids. Think wallpaper paste.
Right. Okay. So once we know we have one of these massive polymers, is there another layer of classification?
Yes, we categorize polysaccharides based on their composition, what they're made of.
Okay.
If they're built from repeating units of only a single type of monosaccharide, say just D-glucose units over and over again. They're called homopolysaccharides.
Homo meaning same.
Exactly. Starch and glycogen are classic examples. They're composed entirely of glucose units, so they're glucans.
And the alternative?
The alternative is heteropolysaccharides, hetero meaning different. These yield a mixture of different sugars or sugar derivatives when you break them down.
And where do we find these? This group includes really crucial components of the body's connective tissue. Things like the mucopolysaccharides, which form the gel-like substance in our joints and skin. We'll definitely talk more about those later.
Okay, let's drill down into the naming system, the grammar, if you like, of those basic monosaccharide units. You said before we even talk 3D shape, we classify them using two key things: the functional group and the number of carbons.
Exactly right. That functional group classification immediately splits all monosaccharides into two main families: aldoses and ketoses.
Aldoses first. What defines them?
And an aldose contains an aldehyde group. Chemically, that's a carbonyl group, C double bond O, situated right at the end of the carbon chain, bonded to at least one hydrogen atom.
Can you give us a quick example?
Sure. The simplest possible aldose is glyceraldehyde. That's a 3-carbon sugar, a triose.
Okay.
And probably the most famous aldose, the primary fuel for our brain, is glucose. Glucose is a 6-carbon sugar, a hexose.
Right. And the other side of that coin, the ketoses.
Ketoses contain a keto group. That means the carbonyl group, the C double bond O, is located internally within the carbon chain. It's usually at the C2 position in most biologically relevant sugars.
An example there.
The simplest ketose is dihydroxyacetone, another 3-carbon triose. But the most prominent ketose in our diet, the one famous for being really sweet, is fructose. That's also a 6-carbon hexose.
Ah, so it's putting these two pieces of information together, the functional group and the carbon count, that gives us the sugar's full, precise name. This is where the naming system, the nomenclature, gets really useful, I imagine.
It's fundamental, absolutely. We use prefixes for the carbon count.
Yeah.
Triose is for 3 carbons, tetrose is for 4, pentose is for 5, hexose is for 6, heptose is for 7, and so on.
So if you tell me glucose is an aldohexose,
You immediately know two things. It's a 6-carbon molecule, hexose, and it carries that reactive aldehyde group at the end of its chain, aldo.
and conversely, fructose is a ketohexose.
Right. 6 carbons again, but this time with the keto group located internally. This chemical language is really the doorway to understanding their potential reactivity and how they behave.
Okay, so we've got the basic classification down. Now, you mentioned earlier that these tiny structural differences are absolutely key. This brings us to the geometry lesson. Stereoisomerism. The idea that you can have two molecules with the exact same list of ingredients, same chemical formula, but because the atoms are arranged differently in 3D space, they're biologically totally distinct.
That's it, exactly.
You used the analogy of keys earlier. Same metal, different cut, fits a different lock.
It's a perfect analogy. Stereoisomerism defines compounds that have the identical structural formula, identical connectivity of atoms, but they differ purely in their spatial configuration. And these differences, they can literally determine whether a molecule is essential food or, just inert material or even toxic. Hashtag 2.1 asymmetry and the isomer formula. Now, the ability of a sugar molecule to even have these different spatial arrangements to form these isomers hinges on one critical feature.
Which is the asymmetric carbon atom. It's often called a chiral center.
And then what makes a carbon asymmetric?
It's a carbon atom that is attached to 4 completely distinct atoms or chemical groups. No two attachments are the same.
Okay. And the number of these asymmetric centers in a molecule tells us something important.
It tells us the potential for complexity. It determines the sheer magnitude of possible structures.
How does that work?
It creates this exponential explosion of possibilities. The number of potential stereoisomers you can have is calculated by a simple formula,
2 to the power of n.
where n is the number of asymmetric carbons in the molecule. Now let's apply that to our main character, glucose. Glucose has 4 asymmetric carbons. C2, C3, C4, and C5 are all chiral.
Okay, so Nollers is 4. 2 raised to the power of 4. $2 times 22 is 4. $4 times 2 is 8. $8 times 22 is 16. 16. That means there are 16 possible sugars that are all isomers of glucose. That's incredible.
It is incredible, and think about the energy the cell saves by being incredibly specific. Nature had 16 potential ways to arrange those exact same atoms for an aldohexose.
Wow.
Yet life, especially mammalian life, relies almost exclusively on just one of those, D-glucose, and maybe a couple of its very close relatives, like D-galactose and D-mannose, which differ only slightly.
The specificity is just astonishing.
It really shows how crucial that exact geometric fit is for our enzymes, for our entire metabolism.
So out of those 16 theoretical possibilities, we only really encounter a handful inside a human cell. And why D-glucose? Why did life settle on that one as the sort of default fuel rather than, say, D-allose or D-glucose?
We really only see a select few in significant amounts. The choice of D-glucose probably boils down to a combination of factors. Its relative metabolic efficiency, perhaps, and the inherent stability of its preferred cyclic structure, its ring form, which we'll definitely get to.
Okay.
But the absolute key takeaway here is that the cell's machinery, all those enzymes, is sculpted over evolutionary time to recognize the exact shape of D-glucose.
So if you somehow ingested one of the other 15 isomers, for the most part, your enzymes simply wouldn't have the complementary shape. They couldn't bind it properly, they couldn't process it efficiently, if at all. It just wouldn't fit the lock. Hashtag tag 2.2 DL configuration. Enantiomers.
Okay, this brings us to the first major distinction in 3D space, the D versus L configuration. You mentioned these are enantiomers.
Yes, enantiomers. That's the technical term for stereoisomers that are non-superimposable mirror images of each other.
Non-superimposable mirror images, like your hands.
Exactly like your hands. Your left hand is a mirror image of your right, but you can't perfectly overlay one on top of the other. They're distinct 3D objects. In biochemistry, this DL distinction is absolutely massive.
So how do we assign whether a sugar is D or L? Is there a reference point?
There is. We use the simplest possible carbohydrate that actually has an asymmetric carbon. That's glyceraldehyde, the 3-carbon aldose. That's our reference standard.
Okay. And what's the specific rule for assigning D or L based on that reference? Because I remember reading that the designation doesn't always tell you which way the molecule rotates light.
That is a crucial point. The DL configuration is purely about the structure, the spatial arrangement, not about its optical activity. They don't always correlate.
So what's the rule?
The rule is based on the position of the hydroxyl group, the OH group, on the asymmetric carbon atom that is furthest from the aldehyde or keto group, but adjacent to the terminal primary alcohol carbon.
Okay, that sounds complicated. For glucose, a hexose, which carbon is that?
For hexoses like glucose, that reference carbon is carbon 5, C5. So you look at the standard way of drawing the molecule flat on paper, the Fischer projection. If the OH group on C5 points to the right, it is defined as a D-series sugar.
T for dextro, right?
Well, historically maybe, but structurally just means right in the Fischer projection. If that C5OH group points to the left, it's an L-series sugar.
L for left, okay. And this single structural definition, right versus left on C5, dictates the entire usability of that sugar for us mammals.
Pretty much, yes. What's the biochemical significance of this overwhelming preference for the D-series? It's arguably the single most important rule in carbohydrate metabolism for mammals.
Why?
Because the vast majority, almost exclusively, the naturally occurring monosaccharides found in mammalian tissues are of the D configuration.
Are we?
Predominantly D. This means our entire enzymatic toolkit, thousands of proteins responsible for breaking down sugars, building them up, using them for signals, is stereospecific. It's tailored like a lock to fit only the D-series sugars.
So if I ate L-glucose,
Your body would essentially treat it as inert material. It can't be phosphorylated by hexokinase, it can't enter glycolysis, it can't be stored as glycogen. It just doesn't fit the enzymes. That single hydroxyl group orientation makes the difference between essential fuel and, well, just something passing through.
okay, so D and L forms are complete mirror images. What about epimers? You mentioned them briefly. They sound like a different kind of subtle difference, like a molecule that's almost identical, but off just at one specific point.
That's an excellent way to put it. Epimers are stereoisomers that differ in their configuration around only one specific asymmetric carbon atom.
Just one.
Just one. And importantly, this definition excludes the anomeric carbon, C1 in aldoses, C2 in ketoses, that has its own special designation we'll get to. So think of it as a very localized structural difference compared to the DL mirror image. But that one difference creates an entirely new, distinct molecule.
We have really key examples of sugars that are metabolically essential, but they only differ from our main guy, glucose, by a single OH flip at one carbon.
Absolutely. The classic examples really highlight how a tiny geometric tweak changes everything. Take glucose and galactose.
Okay.
They are identical except for the orientation of the OH group at carbon 4. So they are C4 epimers.
Just C4.
Just C4. Then take glucose and mannose. They are identical except for the orientation of the OH group at carbon 2. So they are C2 epimers.
C2 epimers. And even though they're so similar, they're treated differently by the body.
Yes. Despite sharing the same chemical formula and most of their structure, they often interact with different transport proteins to get into cells, and they might enter slightly different initial metabolic pathways. The body recognizes them as distinct entities.
So if the body needs, say, galactose to make milk sugar, but mostly we absorb glucose from our diet, how does it handle that? Does it have to make galactose from scratch?
Ah, this is where the cell shows some flexibility. We have specialized enzymes called epimerases.
Epimeresis makes sense.
Their sole job is to catalyze the interconversion between these epimers. This process is called epimerization.
So they can just flip that one OH group.
Essentially, yes. If your mammary gland cell needs galactose to synthesize lactose and all it has readily available is glucose, an epimerase can flip that C4 hydroxyl group on glucose, turning it into galactose. It's like having a little toolkit to modify the building blocks as needed.
That seems much more efficient than building everything from the ground up.
Exactly. It allows for essential shuffling of these closely related structures without requiring completely separate, complex synthetic pathways for each one.
Now, when we talk about these precise structures, C4 epimers, DL forms, it's really easy to visualize them as these flat, straight chains, like we draw them on paper.
The Fisher projections, yes.
But our sources remind us that in the real biological world, which is mostly water, that linear open chain form is actually pretty rare, almost irrelevant structurally speaking.
It's very fleeting. It exists, but only transiently as an intermediate. In solution, monosaccharides are much, much more stable when they curl up and form rings.
They react with themselves.
They do. They're reactive molecules. A hydroxyl group on one part of the carbon chain reacts internally with the aldehyde or keto group at the other end. This forms what's called a cyclic hemiacetal if it started as an aldose, or a cyclic hemiacetal if it started as a ketose.
Okay, so taking glucose again as our main example, which specific carbons are involved in this internal chemical handshake to form the ring?
For glucose, it's the aldehyde group at carbon 1, C1, that reaches across the molecule and reacts with the alcohol group, the hydroxyl group, way down at carbon 5, C5.
C1 reacts with C5.
Yes, this closes the chain into a ring. And for glucose, this forms a particularly stable six-membered ring structure that contains one oxygen atom within the ring itself. This six-membered ring structure is called a pyranose ring.
Pyranose for six-membered. Is there another common size?
Yes, five-membered rings are also common, especially for ketoses like fructose. A five-membered ring is called a furanose ring. Think pyrin versus furan in organic chemistry. Hashtag 3.1 cyclic structure formation, anomers. Now, arguably the most important consequence of this ring closure, the cyclization, is that it creates a new asymmetric center in the molecule.
A new one.
Or at C1 for aldoses like glucose. Remember, in the open chain form, C1 was the aldehyde carbon. It was double bonded to oxygen and bonded to hydrogen. So it only had 3 different attachments, not 4. It wasn't chiral. But when it forms the hemiacetal in the ring, that C1 carbon becomes bonded to 4 different things. The oxygen from C5, the original hydrogen, the rest of the ring, C2, and a new hydroxyl group formed during the reaction. It's now chiral.
Ah, okay. And this new chiral center has a special name.
It does. We call this new asymmetric center, created by a ring formation, the anomeric carbon.
Anomeric carbon. C1 in glucose?
C1 in glucose, yes. Or C2 in ketoses like fructose. And because this anomeric carbon is now chiral, it can exist in 2 different spatial configurations.
And these are the alpha and beta forms we hear about.
Exactly. These 2 resulting cyclic structures, which differ only in the configuration around that newly formed anomeric carbon, are called anomers.
Alpha and beta anomers.
Yes. And the distinction is purely spatial, how that new OH group on the anomeric carbon is oriented relative to the rest of the ring. In the standard way we draw glucose rings, here with projections, in the alpha anomer, the OH group on C1 typically points down or axially. In the beta anomer, the OH group on C1 typically points up or equatorially. Another way to define it, which works regardless of drawing convention, is relative to the CH2OH group, C6, that sticks out of the ring. In the alpha anomer, the C1OH is on the opposite side of the ring from the C6. In the beta anomer, the C1OH is on the same side as the C6 group.
It seems like such a tiny difference. Does it really matter?
It matters immensely because that anomeric hydroxyl group is the one that will participate in forming glycosidic bonds to link sugars together. Whether it's in the alpha or beta position dictates the shape of the resulting bond and the larger molecule, like starch versus cellulose. It's absolutely critical.
Okay, so we have these 2 anomers, alpha and beta. And you said the straight chain form is just a fleeting intermediate. The fact that these forms exist and can sort of flip between each other through that intermediate leads to this phenomenon called mutarotation, right?
Yes, exactly.
This is the actual experimental evidence that sugars aren't just static rings in water, but they're constantly dynamically opening and closing.
Mutarotation is the perfect demonstration of the dynamic nature of sugars in solution. It's defined as the change in specific optical rotation that occurs when you dissolve a sample of a pure anomer, either pure alpha or pure beta, in water.
Optical rotation. That's how the sugar solution bends polarized light.
Precisely. Because stereoisomers interact differently with plain polarized light, the alpha and beta anomers have different initial optical rotation values. But as they sit in solution, they start to interconvert. The rings open briefly to the linear form and then reclose, sometimes as alpha, sometimes as beta.
So the mixture changes over time.
The mixture changes until it reaches a stable equilibrium, a constant ratio of alpha and beta forms. And since the alpha and beta forms rotate light differently, we can actually measure this shift in the overall rotation of the solution as it reaches that equilibrium.
Can you walk us through the actual numbers for D-glucose that makes it clearer?
Sure. Let's look at the specific data points from the sources. If you take a sample of pure crystalline alpha-2-glucose and dissolve it in water, its initial specific optical rotation is quite strongly positive, plus 112 two-words.
Okay, plus 112.2.
Now, if you take a sample of pure crystalline beta-glucose and dissolve that, its initial rotation is much lower, still positive, but only plus 18.7 to pound.
Big difference. Plus 18.7.
Huge difference. But here's the key. Regardless of whether you started with pure alpha or pure beta, if you let that solution sit, the optical rotation will gradually change over time. It will drift
Until
until it stabilizes at a single constant weighted average value, plus $52.07.
always plus 52.7 degrees for glucose.
For D-glucose at equilibrium, yes, that stable value represents the final relaxed state of the system, the equilibrium mixture.
And you said this interconversion happens via that open chain form.
Yes, the ring has to open briefly into the linear aldehyde form, and then it can reclose into either the alpha or beta ring. But that linear form is really insignificant concentration-wise. Maybe only 1% or less of the molecules are in that form at any given instant.
Right. And at equilibrium, is it a 50-50 mix of alpha and beta?
No, it's not. The beta anomer is actually slightly more stable, thermodynamically favored, in aqueous solution. Part of the reason is that its bulky OH group on C1 is often in the more spacious equatorial position in the chair conformation, leading to less steric hindrance.
Okay, so beta's more stable. What's the ratio then?
So at equilibrium, the beta anomer predominates. The final mixture is about 63% beta glucose and about 36% alpha D glucose, with that tiny trace of the open chain form making up the rest. The plus 52.7 degrees is the weighted average rotation of that specific mixture.
Fascinating. And we see the same dynamic opening and closing, this mutarotation, in ketones like fructose too. You mentioned fructose can form different ring sizes.
Absolutely. Fructose also undergoes mutarotation, and it's structurally even more flexible, you could say. While glucose strongly prefers that six-membered pyranose ring, fructose readily interconverts between the pyranose, six-membered, and the furanose, five-membered ring forms in solution.
So it's even more dynamic.
In a way, yes. This conformational flexibility, being able to adopt different ring shapes, is thought to be one reason why fructose tastes significantly sweeter than glucose. Different receptors in our taste buds might recognize and bind to these different forms. When fructose reaches its equilibrium in solution, its final rotation stabilizes at 92 testor.
Minus 92, so it rotates light strongly to the left.
Yes, fructose is strongly levorotatory, hence its other name, levulose. Glucose, being dextrorotatory, is also called dextrose. Hashtag 3.3, reducing properties and interconversion.
Okay, so that anomeric carbon, C1 in glucose, the one that's formed during ring closure and is involved in mutarotation,
Yeah.
that's also the key to another major property of sugars, their ability to act as reducing agents.
Exactly. That anomeric carbon, specifically its ability to revert back to the open chain form with its free aldehyde or keto group, is what gives many frizzlers their chemical superpower, as you called it.
The power to reduce things. Why is that important?
It's historically been vital for detecting and quantifying sugars, like in the Benedict's test for glucose in urine. The property is especially pronounced when the sugar is in an alkaline or basic solution.
What happens in alkaline conditions?
Under alkaline conditions, sugars undergo a critical structural rearrangement called tautomerization. It involves the migration of a hydrogen atom and a shift in the position of a double bond.
Okay, Polymerization. What does that produce?
This process generates highly reactive intermediate structures called enedials. Enia for the double bond, diol for two hydroxyl groups attached to the double bonded carbons.
Enediols, and these are the reactive species.
Yes, these enedials are unstable and very readily oxidized. Because they get oxidized so easily, they can act as potent reducing agents. They donate electrons to other molecules, like the copper ions in Benedict's region, reducing the copper from blue C2 plus to reddish Q plus Le.
And this ability is directly linked back to that anomeric carbon being able to open up.
Directly. If the anomeric carbon is locked, say in a glycosidic bond, like in sucrose, the ring can't open, no enediol can form, and the sugar is non-reducing. So we call sugars with a free or potentially free aldehyde keto group reducing sugars.
Now, this reactive enediol intermediate, that's also the explanation for something quite surprising. The fact that three major sugars, glucose, fructose, and mannose, can actually turn into each other under certain lab conditions.
Yes, that's the Lobry-de Broglie-von-Ekenstein transformation. It sounds complicated, but the core idea relies on that shared enediol intermediate.
How does that work? How does a shared intermediate bridge the structural gaps between glucose, fructose, the ketose, and mannose, a C2 epimer of glucose?
Okay, think about their structures near the top. Glucose and mannose differ only at C2. Fructose is a ketose, so its structure is different at both C1 and C2 compared to glucose. However, when any of these three sugars are placed in mild alkaline conditions, they all tautotomerize to form the same 1-gover-2 enediol intermediate.
They all converge on the same intermediate structure.
For that C1-C2 region, yes. The double bond forms between C1 and C2, and the stereochemistry at C2 is temporarily lost. Since they all share this common intermediate structure, the process is reversible. The enediol can collapse back down, reforming the carbonyl group, but it can resolve into any of the three starting sugars.
So you can start with pure glucose and alkali and end up with a mixture of glucose, fructose, and mannose.
Exactly. It's an equilibrium process. This transformation really underscores just how dynamic and somewhat fluid the structural distinctions between these closely related sugars can be under specific chemical conditions.
And that explains the really interesting results you get with a specific chemical test called osazone formation.
It connects directly to that, yes. The osazone test involves reacting a sugar with an excess of a reagent called phenylhydrazine. This reaction specifically involves carbons 1 and 2 of the sugar.
C1 and C2 again.
Right. Since the structural differences between glucose, fructose, and mannose only exist around C1 and C2, and since these carbons become heavily modified and essentially masked by reacting with 3 molecules of phenylhydrazine.
The final product ends up being the same for all three.
Precisely. Glucose, fructose, and mannose all yield the exact same osazone crystals, which are characteristically needle-shaped. This is strong chemical evidence that their structural differences are localized to that C1-C2 region, the same region involved in the enediol interconversion. Galactose, which differs at C4, forms a different shaped osazone.
Okay, we've really dug into the complexity of a single sugar unit, the isomers, the rings, the anomers, the reactivity. Now let's move to linking them together. Polymerization. When two or more monosaccharides join up, they form what's called a glycosidic bond. And the formation of this bond seems crucial because it completely locks in that alpha or beta configuration at the anomeric carbon that we just spent time detailing.
Absolutely. The glycosidic bond is the fundamental covalent linkage that builds up all the complex carbohydrates, from simple disaccharides to massive polysaccharides like starch and cellulose.
How does it form?
It typically forms when the hydroxyl group on the anomeric carbon, C1 or C2 of one sugar, reacts with any hydroxyl group on another molecule.
It doesn't have to be another sugar.
No, often it is, linking sugar to sugar. But that second molecule could also be a non-carbohydrate component, like an alcohol, a phenol, or even a nitrogen atom in an amine. If the second part isn't a sugar, we call it an aglycone. The whole molecule is then called a glycoside. Hashtag 4.1, the glycosidic bond and nomenclature. But the absolutely critical point, getting back to anomers, is the specific stereochemistry, whether the reacting anomeric carbon was in the alpha or beta Collocker configuration before the bond formed. Because once that bond forms, the ring is locked. It can no longer open and close at that position.
So that alpha or beta designation becomes permanent in the linkage.
It becomes a permanent structural feature of the bond itself. And this dictates everything, the shape of the resulting molecule, which enzymes can recognize and break that bond, etc.
So when we see chemists or biologists write out a linkage like, say, beta 1 right RO4R, that notation isn't just shorthand. It's a precise structural instruction manual.
It tells you everything you need to know about that specific connection. The nomenclature of the glycosidic bond is incredibly informative. Let's break down the beta-right-RO4
Yep.
The clay printout tells you it involves the anomeric carbon C1 of the first sugar. The 4 tells you it connects to the hydroxyl group on C4 of the second sugar. And the beta tells you the crucial stereochemistry. The anomeric carbon of that first sugar was in the beta configuration when the bond formed.
So just 3 symbols tell you the connection points and the 3D orientation.
Exactly. An alpha-1 right RO4 bond, like in starch, is fundamentally different geometrically from a beta right RO4 bond, like in cellulose.
And this structural lock, formed by the glycosidic bond, is also what determines whether the resulting disaccharide is classified as reducing or non-reducing, which you mentioned earlier.
Correct. It depends on which hydroxyl groups are involved. If a disaccharide is formed such that one of the original monosaccharide units still has a free anomeric carbon, meaning its ring can still open up to potentially expose the aldehyde or keto group, then the whole disaccharide remains a reducing sugar. Maltose and lactose are examples.
Okay.
But if the glycosidic bond is formed between the anomeric carbons of both sugar units, then neither ring can open up. Both potentially reducing groups are locked in the bond. In that case, the molecule becomes a non-reducing sugar. Sucrose is the classic example. It can't form anidiols, can't reduce copper ions. Hashtag tagged 4.2 biochemically important disaccharides.
Right, let's quickly review the 3 giants of the disaccharide world, highlighting these features, starting with maltose or malt sugar. We'll get this from breaking down starch, right?
Yes, maltose is essentially 2 glucose units stuck together. Specifically, it's 2 alpha-D glucose units.
And the linkage.
They are linked by an alpha-1 right RO4 glycosidic bond. So the anomeric C1 of the first glucose in its alpha form connects to the C4 hydroxyl of the second glucose.
Okay, alpha-1 rido 4, and is it reducing?
Yes, because the anomeric carbon C1 of the second glucose unit is not involved in the bond, it's free. So that ring can still open and close, meaning maltose is indeed a reducing sugar.
When if you did the ozazone test on it.
Maltose yields characteristic, beautiful sunflower-shaped ozazone crystals, different from the needle shape of glucose-fructose monos.
Okay, next up, lactose, the famous milk sugar critical for babies.
Lactose is structurally distinct. It's composed of a beta-D galactose unit linked to a D-glucose unit. Remember, galactose is the C4 epimer of glucose.
Right, and the linkage here.
The bond is a beta-1 RO4 glycosidic bond. So the anomeric C1 of the galactose in its beta form connects to the C4 hydroxyl of the glucose.
Say 1,4 this time. Reducing or non-reducing?
Still reducing. Again, the anomeric C1 of the glucose unit is free and available to open up.
Makes sense.
And the sources really emphasize its fundamental importance in nutrition, noting it's the most crucial carbohydrate source for young mammals. If you test lactose, it yields powder puff, or sometimes described as hedgehog-shaped ozozones.
Powder puff. Okay, and finally, sucrose. Ordinary table sugar. You said this one is the structural anomaly in the family.
Sucrose is quite unique because it links an aldose, glucose, with a ketose, fructose. Specifically, it's formed from alpha-D glucose and beta-D fructose.
Alpha glucose and beta fructose, how are they linked?
The linkage is highly specific. It's an alpha-1 right arrow beta-2,2 glycosidic bond. This means the anomeric carbon of glucose, C1 in alpha form, is linked directly to the anomeric carbon of fructose, C2 in beta form.
Ah, so it links anomeric carbon to anomeric carbon?
Exactly. The potentially reducing groups of both sugar units are tied up, locked into that glycosidic bond.
And since both reducing sites are locked, the molecule has no way to open up, no way to form an anediol.
Correct. Sucrose is therefore a completely non-reducing sugar, which also means diagnostically it cannot form ozozones. That's a key chemical difference setting it apart from maltose and lactose.
And this unique lock structure is also directly responsible for that phenomenon you mentioned earlier called inversion.
Yeah.
It sounded complicated, but you said it's really just a trick of light rotation upon breaking the bond.
It is. It's a rather striking laboratory observation, though. Pure sucrose in solution is dextrorotatory. It rotates plain polarized light to the right. The specific rotation is plus $66.05.
Okay, positive rotation.
But when you hydrolyze sucrose, break that alpha-1 right arrow beta-2,2 bond using either acid or a specific enzyme called invertase or sucrase, you release a 1-to-1 mixture of free glucose and free fructose.
Right.
Now we know glucose at equilibrium is dextrorotatory plus $52.07. But fructose, as we established, is strongly levorotatory, 92 test.
Ah, so the negative rotation of fructose is much stronger than the positive rotation of glucose.
Exactly. The strong negative rotation of the liberated fructose dominates the mixture. The resulting one-to-one mix of glucose and fructose, called invert sugar, has an overall negative specific rotation around $28.02.
So the rotation flipped from positive to negative.
It inverted, hence the name inversion for the hydrolysis process and invert sugar for the resulting mixture. It's just a consequence of the optical properties of the constituent monomers versus the original disaccharide.
Okay, great. We've covered the dimers. Now let's shift focus fully to the really big polymers, polysaccharides. We're moving from these relatively small, often soluble sweet structures to these huge, often dense molecules that serve, as you said earlier, two main kinds of roles, either massive energy reserves or physical scaffolding and fortification.
That's a perfect summary of their major functions, yes. Hashtag tag 5.1 homopolysaccharides, storage and fuel. Let's start with the storage ones, built from a single type of monomer homopolysaccharides. And the absolute king here, certainly in plants and in our diet, is starch.
The most important carbohydrate we eat, basically.
Arguably, yes. It's the primary way plants store energy. Structurally, it's a polymer built entirely of D-glucose units, making it a glucan. But starch isn't actually one single substance. It's typically a mixture of two different glucan structures.
Two components in starch, what are they?
There's amylose, which is essentially a long, linear, unbranched chain of glucose units linked purely by alpha-1 right arrow 4 glycosidic bonds. This structure tends to coil up into a helix.
Okay, amylose is the straight chain.
Mostly straight chain, yes. The other component is amylopectin. This also has a backbone of alpha-1 right arrow 4 linked glucose units, but is highly branched.
Branched how?
At various points along the alpha-1 right arrow 4 chain, additional short chains branch off, connected via alpha-1 right arrow 6 glycosidic bonds. So amylopectin is this big, bushy molecule compared to the linear amylose. Most starches are predominantly amylopectin.
Got it. And what about the equivalent energy storage molecule in animals?
That would be glycogen. It's often referred to as animal starch because its function is analogous. It's a readily accessible glucose reserve.
Where do we store it?
Primarily in the liver, where it helps maintain blood glucose levels, and in muscle tissue, where it provides fuel for muscle contraction.
And structurally, how does glycogen compare to starch?
Structurally, glycogen is very similar to amylopectin. It's also a branched glucan with alpha-1 right arrow 4 more backbones and alpha-1 right arrow 6 level branch points. The main difference is that glycogen is even more highly branched than amylopectin.
More branches. Why is that important?
That high degree of branching is functionally crucial. It creates many non-reducing ends on the molecule. Enzymes that break down glycogen, like glycogen phosphorylase, work from these ends. So having lots of branches means lots of ends for enzymes to attack simultaneously.
Which means faster glucose release.
Much faster mobilization of glucose units to meet sudden immediate energy demands, like during exercise or stress. It's a very efficient structure for rapid fuel delivery.
Okay. Now, we have to briefly mention inulin here. You brought it up earlier. It's a homopolysaccharide, but its importance isn't as fuel, but rather as a diagnostic tool.
That's right. Inulin is unique among the storage polysaccharides we usually discuss because it's a polymer of fructose, not glucose. So it's a fructosan or fructan.
A chain of fructose units.
Yes. And the key thing is, the human body lacks the enzymes needed to hydrolyze the specific glycosidic bonds in inulin. We can't digest it, we can't metabolize it.
So it just passes through us.
Essentially, yes.
Yeah.
It gets filtered by the kidneys but isn't reabsorbed or secreted by the renal tubules. This inertness, this predictable filtration, makes it incredibly useful clinically for assessing kidney function. Doctors can inject a known amount of inulin into the bloodstream and then measure how quickly it appears in the urine and its concentration. Because it's cleared solely by glomerular filtration, this measurement allows for a very precise determination of the glomerular filtration rate, or GFR, a key indicator of overall kidney health. Hashtag 5.2 homopolysaccharides, structural roles.
Okay, we've covered energy storage. Now let's switch to the homopolysaccharides that provide rigid physical structure. You mentioned cellulose in plants and chitin in invertebrates earlier, and you hinted that the structural difference between soft starch and hardwood is incredibly tiny chemically.
It's probably the most dramatic illustration in all of biochemistry of how a subtle difference in stereochemistry dictates a massive difference in physical properties and biological function. Let's look at cellulose.
Main component of plant cell walls, right? Wood, cotton fiber.
Exactly. It's the most abundant organic polymer on Earth. Structurally, like starch, it is also a polymer of D-glucose units, a glucan.
Okay, so far sounds the same as starch. What's the difference?
The only chemical difference lies in the glycosidic linkage between the glucose units. Instead of the alpha-1 right-arrow link found in starch and glycogen, cellulose utilizes the beta-arrow glycosidic bond.
Wait, that's it. The only difference between digestible helical starch and rigid indigestible wood fiber is the orientation of that bond at C1, alpha versus beta.
That is the entire difference at the chemical linkage level. It's almost unbelievable, but it's true.
How does that one change cause such a massive difference in properties?
The alpha linkage in starch allows the polymer chain to adopt a flexible, coiled, helical conformation. This structure is relatively accessible to water and, crucially, to the digestive enzymes, amylases, in our saliva and gut that are designed to break alpha-1 right-O4 Gall bonds.
Okay. And the beta linkage.
The beta linkage in cellulose forces the glucose units into an extended linear chain conformation. These straight chains then align parallel to each other, forming extensive networks of hydrogen bonds between adjacent chains.
So they pack together really tightly.
Incredibly tightly, forming strong, rigid microfibrils. This structure provides immense tensile strength, perfect for a plant cell wall, but it's also very resistant to chemical attack. And critically, mammals, including humans, completely lack the necessary enzyme called cellulase that can specifically cleave that tough beta-ritoforward glycosidic bond.
So we simply can't digest it. It's inedible for us.
Nutritionally, yes, we get no calories from cellulose.
But despite being indigestible, our sources still highlight it as a crucial part of a healthy diet. Why?
Because it forms the bulk of what we call dietary fiber. While it provides no energy, it plays essential physiological roles in the digestive tract.
Such as?
Fiber adds bulk to the intestinal contents, which helps stimulate peristalsis, the muscle contractions that move food along. It can also slow down the rate at which glucose from digestible carbohydrates is absorbed, helping to moderate blood sugar spikes after a meal. Some fibers can bind cholesterol and bile acids, aiding their excretion, so it's highly functional for gut health, even though it's not strictly nutritional.
Functional without being fuel.
Yeah.
Got it. And the other major structural homopolysaccharide you mentioned was chitin. What's its role?
Chitin serves a very similar structural purpose, but primarily in the animal and fungal kingdoms. It's the main structural polysaccharide found in the tough, protective exoskeleton of invertebrates. Think of the shells of insects, crabs, lobsters. It's also found in the cell walls of fungi.
So it's like the cellulose of the invertebrate world. What's it made of?
It's structurally very similar to cellulose. It's also a long, unbranched polymer linked by beta-1 right-RO4 glycosidic bonds. The difference is the monomer unit. Instead of glucose, chitin is a polymer of N-acetyl-D-glucosamine, which is essentially glucose with an acetylated amino group attached at C2. That modification doesn't change the overall structural principle of forming rigid fibers via beta linkages. Hashtag tag 5.3 heteropolysaccharides, the tissue matrix.
Okay, we've covered the rigid structures made from single sugar types. Now let's talk about biological materials that need to be more flexible, maybe elastic or shock absorbent or lubricating. This brings us to the other class of polysaccharides, the heteropolysaccharides, the ones made from different repeating units. And the major group here seems to be the glycosaminoglycans, or GAGs.
Yes, glycosaminoglycans, or GAGs, are absolutely fundamental to the structure and function of our connective tissues. They were also historically called mucopolysaccharides because they often form viscous, mucus-like solutions.
And these aren't about rigidity like cellulose.
No, their role is quite different. They typically form the essential, hydrated, gel-like ground substance or extracellular matrix, ECM. This is the stuff that fills the spaces between cells in tissues like cartilage, skin, tendons, and blood vessels. It's the material within which the fibrous proteins like collagen and elastin are embedded.
So they provide the supportive, squishy environment. What are they made of?
They are typically long, unbranched chains composed of repeating disaccharide units. And crucially, these repeating units almost always contain an amino sugar, like N-acetylglucosamine or N-acetylgalactosamine, and often an acidic sugar, like glucuronic acid. Many GAGs are also heavily sulfated, meaning they carry sulfate groups.
Amino sugars, acidic sugars, sulfates. It sounds like they carry a lot of negative charge.
They do. They're highly negatively charged polymers. This high charge density causes them to repel each other and attract large amounts of water, forming that hydrated space-filling gel which resists compression, perfect for rolls in joints and cartilage.
Right. Let's look at some key GAG examples from the sources that perform these really specialized non-fuel roles, starting with hyaluronic acid.
Hyaluronic acid, or hyaluronan, is a bit unique among GAGs as it's non-sulfated and incredibly large. It's a major component of synovial fluid, the fluid that lubricates our joints. Its high viscosity makes it an excellent lubricant and shock absorber, cushioning bones during movement.
Okay, joint lubrication, any other roles?
Yes, fascinatingly, it plays a completely different role in reproduction. It forms a thick, gel-like matrix around the ovum, the egg cell. For fertilization to occur, sperm must penetrate this barrier. They do so by releasing an enzyme called hyaluronidase, which specifically breaks down hyaluronic acid.
Wow, so it's both a mechanical cushion and a kind of reproductive gatekeeper, versatile molecule.
Incredibly so.
Okay, what about some other key gags? You mentioned some have roles in blood or tissue support.
Yes. Heparin is probably the most well-known JAG clinically. It's produced by mast cells and is found lining blood vessels. Its major function is as a potent natural anticoagulant. It strongly inhibits blood clotting, which is why it's widely used medically to prevent or treat blood clots.
An anticoagulant built from sugars,
Okay.
and for tissue shape and structure.
Then we have the chondroitin sulfates. These are major components of cartilage, bone, skin, tendons, and ligaments. They are crucial for maintaining the structural integrity and shape of these tissues, largely due to their ability to bind water and resist compressive forces. They literally help give tissues their resilience and form.
Makes sense for cartilage especially. And finally, the sources mentioned one critical for vision.
That's keratin sulfate. This JAG is particularly important in the structure of the cornea, the transparent outer layer of the eye. It also contributes to bone and cartilage structure. Its specific arrangement and hydration properties within the corneal tissue are essential for maintaining the transparency of the cornea.
Transparency relies on a sugar structure.
Absolutely. If the structure or amount of keratin sulfate is abnormal, as happens in some genetic conditions, the cornea can become cloudy, severely impairing vision. It really drives home the point. These complex carbohydrates are essential for life's most intricate functions, far, far beyond just providing energy.
All right, we're moving now beyond the big structural and storage roles into even more specialized functions of carbohydrates and their derivatives, how they interact with our genetics, our immune system, and other really specific physiological processes. Hashtag tag 6.1 genetic structure and metabolic intermediates.
Well, you can't get much more specialized or fundamental than the building blocks of our genetic material. The 5-carbon sugar D-ribose is an absolutely critical component of RNA, ribonucleic acid, the molecule involved in translating genetic information into proteins.
Ribose and RNA.
Okay, but it doesn't stop there. Ribose is also the core sugar component of several essential nucleotide coenzymes. When we talk about the cell's energy currency, we mean ATP, adenosine triphosphate. The A involves adenine, but the TP part is linked via ribose.
So the energy carrier itself is built on a sugar.
Yes. And the same goes for the crucial electron carriers in metabolism. NAD+, nicotinamide adenine dinucleotide, and NADP+, its phosphate version. Ribose forms the structural backbone connecting the nicotinamide and adenine parts in both of these essential redox cofactors.
Wow, so ribose is central to RNA, ATP, NAD plus NADP.
Yeah,
Pretty vital stuff.
absolutely fundamental.
And the sugar in DNA is just a slight modification of ribose, right?
Exactly. The structural constituent of DNA, deoxyribonucleic acid, is D2-deoxyribose. As the name implies, it's simply ribose that is missing the oxygen atom at the C2 position.
Deoxy. Got it.
That single oxygen atom difference is profound. It contributes to the greater stability of DNA compared to RNA, making DNA better suited for long-term storage of genetic information, while RNA handles the more transient information transfer and catalytic roles. Again, a tiny structural change dictates a massive functional divergence.
It's a recurring theme. And even within the active metabolic pathways themselves, like when we're breaking down glucose, sugars exist as intermediate forms, not just the start and end points.
Constantly. Monosaccharides and their phosphorylated derivatives are the bread and butter of central metabolism. In glycolysis, the main pathway for glucose breakdown, we see crucial intermediates like glucose 6-phosphate, fructose 6-phosphate, fructose 1006-bisphosphate, and glyceraldehyde 3-phosphate. These aren't just fleeting steps, they are key regulatory points and branch to other pathways.
And other pathways too.
Yes, for instance, the hexose monophosphate shunt or pentose phosphate pathway, which is crucial for generating NADPH, needed for biosynthesis and combating oxidative stress, and producing ribose for nucleotides. That pathway involves specialized sugar phosphates like D-ribulose 5-phosphate and D-cetoheptulose 7-phosphate, a 7-carbon sugar. These intermediates are the chemical currency being passed along the metabolic assembly lines. Hashtag 6.2 cellular recognition and immunology, glycoconjugates. So if the inside of the cell is using sugars primarily for energy and as metabolic building blocks, the outside of the cell often uses them for communication and recognition.
How does that work?
This happens when carbohydrates are covalently attached to other biomolecules, primarily proteins or lipids, forming structures called glycoconjugates. If a carbohydrate chain, lichen, is attached to a protein, it's a glycoprotein. If it's attached to a lipid, it's a glycolipid.
And these glycoconjugates are often found where?
They are major components of the outer surface of cell membranes, with the carbohydrate portions typically extending outwards from the cell surface into the extracellular environment.
So they act like little flags or antennas on the cell surface.
That's a great analogy. These carbohydrate chains mediate a huge range of crucial cellular interactions. They're involved in cell growth regulation, how cells adhere to each other to form tissues, cell adhesion, and specific recognition events like how sperm recognizes an egg during fertilization.
And they're important in the immune system, too.
Hugely important. Many key proteins involved in immunity are glycoproteins. Think of immunoglobulins, which are antibodies, the Y-shaped proteins that recognize and neutralize pathogens. The carbohydrate portions on antibodies are critical for their stability and function. Other glycoproteins like fibronectin and laminin, which are components of the extracellular matrix, are vital for cell recognition, adhesion, and guiding cell migration during development and wound healing. They help define tissue boundaries and maintain tissue integrity.
Okay. But probably the most famous and easily understood example of carbohydrates defining our identity is through blood groups, right?
Absolutely. The ABO blood group substances, the antigens found on the surface of your red blood cells that determine whether you are type A, B, AB, or O, are classic examples of glycoconjugates. They can be other glycoproteins or glycolipids.
And the difference between A, B, and O is purely down to the sugar part.
Purely down to the specific structure of the short carbohydrate chain, oligosaccharide, that's attached. All blood types share a common core oligosaccharide structure. Type O individuals have just that core structure. Type A individuals have an enzyme that adds an extra N-acetylgalactosamine sugar to that core. Type B individuals have a slightly different enzyme that adds an extra galactose sugar instead. Type AB individuals have both enzymes, so they add both sugars.
So a single sugar difference determines your blood type and who you can safely receive blood from.
Exactly. It's the ultimate example of how a very specific carbohydrate structure dictates individual immunological identity and has major clinical consequences. Hashtag 6.3 unusual in clinical functions.
We're almost at the end. But the sources highlight a few more fascinating, sometimes unusual, and clinically relevant roles for sugars and their derivatives. Things that seem completely removed from basic energy or structure.
Yes, there are some really specialized cases. Let's start with the darker side. Pathology linked to sugar derivatives.
Okay.
In certain conditions, particularly uncontrolled diabetes where blood glucose is consistently very high, some cells start shunting excess glucose into alternative metabolic pathways. One such pathway converts glucose into a sugar alcohol called sorbitol and galactose into dulcidol.
Sugar alcohols, what's the problem with them?
The problem is that these sugar alcohols are not easily metabolized further in some tissues, like the lens, including cataract formation in the lens, peripheral neuropathy, nerve damage, and nephropathy, kidney damage.
Wow, so sugar accumulation causing physical damage through osmosis. Okay. Now, on the flip side, on the therapeutic side, the sources mentioned a synthetic sugar being used to treat a specific liver-related disorder.
Yes, that's lactulose. It's a synthetic disaccharide made from galactose and fructose, linked in a way that human intestinal enzymes cannot break down.
So, like inulin and cellulose, it's not absorbable for us.
Correct. Because it's not absorbed in the small intestine, it travels intact to the large intestine, the colon. There, resident bacteria can ferment it.
And what does that achieve?
Lactulose is used primarily to treat hepatic encephalopathy. This is a serious neurological condition caused by advanced liver failure, where the liver can no longer effectively detoxify ammonia produced in the gut. High ammonia levels in the blood are toxic to the brain.
So how does lactulose help?
When bacteria in the colon ferment lactulose, they produce acidic byproducts like lactic acid and acetic acid. These acids lower the pH of the colon. In this more acidic environment, the toxic ammonia readily picks up a proton, H+, and gets converted into the ammonium ion, and H3 becomes NH4 plus SAR.
NH3 becomes NH4 plus mighty. Why is that better?
Because the ammonium ion, Sexton H4 plus an Iris charge, it is much less readily absorbed across the intestinal wall back into the bloodstream compared to the uncharged ammonia. So the ammonia gets trapped in the colon as ammonium and is then simply excreted in the feces. It's a clever way to lower toxic blood ammonia levels using a non-absorbable sugar.
That is clever. Trapping the toxin using acidity generated from sugar fermentation. Okay, finally, let's just quickly revisit those two really cool examples that highlight the extreme versatility of sugar chemistry, one from nature, one from medicine.
Right. First, the sheer evolutionary ingenuity seen in certain Antarctic fish. They survive in seawater that's actually below the normal freezing point of water. How? They produce specialized antifreeze glycoproteins.
Antifreeze made of sugar and protein. How does it work?
These molecules don't lower the freezing point significantly like salt does. Instead, they work by adsorbing or binding onto the surface of any tiny ice crystals that start to form in the fish's body fluids. By binding to the crystal lattice, they physically prevent the crystals from growing larger. It's a non-colligative mechanism, inhibiting ice growth rather than lowering the freezing point overall. It's carbohydrate chemistry acting as nature's sophisticated survival tool.
Amazing. And the second example was antibiotics.
Yes. Many important antibiotics are actually glycosides, meaning they have a sugar component attached to a non-sugar aglycogen part. Our sources specifically cite streptomycin as a classic example. Streptomycin is an aminoglycoside antibiotic, effective against certain types of bacteria. The complex carbohydrate portion is absolutely integral to its mechanism of action, which involves interfering with bacterial protein synthesis. It shows that complex sugars can be powerful weapons in the fight against infection. Hashtag outro.
So where does this leave us? We started this deep dive thinking about carbohydrates as, simple fuel, counting calories from bread or sugar. And we end up having seen them as the architects of wood and cartilage, the communication flags on our cell surfaces, the literal backbones of our genetic material, and even as molecular antifreeze or life-saving drugs. It's quite a journey.
It really is. I think the biggest takeaway is that life, at its most fundamental level, is built upon the most precise, intricate molecular geometry. We've navigated almost the entire carbohydrate spectrum today. From that basic DL distinction that determines if a sugar is even useful to us metabolically, to the subtle difference between an alpha and a beta linkage at C1 that dictates whether a glucose polymer becomes digestible fuel, like starch, or indigestible structural fiber, like cellulose.
Every single role seemed to hinge on knowing exactly where each atom, especially those hydroxyl groups, was pointing in 3D space.
Exactly. Think about the lubricant function of hyaluron required of keratin sulfate. These highly specialized functions all depend utterly on the precise derived from that specific sugar structure.
And that profound specificity brings us right back to where we started, really. That almost absolute preference our bodies show for D sugars over L sugars.
It does. We emphasize that our entire complex enzyme machinery built up over millions of years of evolution is exclusively dedicated to recognizing and processing D sugars.
It's an absolute uncompromising preference.
It really is. And maybe the final thought to leave you with is just to ponder the sheer efficiency and perhaps vulnerability of that. Imagine if you could magically synthesize a batch of L-glucose, the perfect mirror image of our normal fuel, and you consumed it.
My body would just ignore it.
It would be functionally invisible to your metabolic pathways. The most basic, fundamental energy pathway that powers your brain, your muscles, the pathway that arguably enabled all complex life to evolve. It hinges entirely on the specific spatial orientation of one single hydroxyl group on one specific carbon atom, C5. This simple yet profound geometrical law is truly the secret life of sugar. Welcome back to the deep dive. Today we are undertaking a deep dive into a group of molecules that are, well, arguably just crucial components of life, yet often get dismissed with just one word: fat. We're going beyond those, dietary ideas to explore lipids, these highly concentrated energy sources, the essential signaling messengers, and the tireless architects of every single cell boundary we have.
That's absolutely right. Yeah, our stack of source material today gives us a really comprehensive look at what makes this group of organic substances so, well, indispensable.
Yeah.
They're incredibly diverse chemically, functionally, which makes them honestly a bit tricky to study sometimes.
Right.
So our mission is pretty clear. We're going to define lipids precisely, try to impose some structure on their complex classification, and really unlock the core biochemistry that dictates their structural and regulatory power.
Okay, let's unpack this then with the absolute fundamental definition. When we talk about lipids, we're not talking about a class defined by, say, chemical structure, like proteins are. We're defining them by specific behavior. What is that essential defining characteristic?
It really all comes down to their relationship with water. Lipids are organic substances characterized by their relative, but pretty dramatic, insolubility in water. They're hydrophobic. And conversely, their solubility in non-polar organic solvents like alcohol, chloroform, or ether. This distinction is absolutely critical.
They hate water.
They literally shun aqueous environments, yeah.
Our source material captures that personality perfectly, describing the lipid's attitude toward water with the quote, "With water, I say, touch me not." That hydrophobic behavior is really the key to understanding all their functions, isn't it?
It truly is. And this brings up another really critical differentiating factor. Unlike proteins or polysaccharides or nucleic acids, lipids are not polymers. They aren't built from repeating monomer units.
Okay.
They're typically smaller molecules. And this structural difference allows them to serve.
Yeah.
First, they're the chief concentrated, like the powerhouse. And second, they are the fundamental structural components that form the boundaries and internal scaffolding of our cells. They are the highly efficient and the precise building code, all wrapped up.
That sets the perfect framework for what we'll cover because the sheer chemical, heterogeneity of lipids ranging from simple oils to complex hormones, it really requires a systematic approach. We need to classify this family. Our sources rely on a modified system, often linked to Bloor, which divides them into 4 major classes. Let's start with the most basic category, simple lipids.
Okay, simple lipids are, chemically speaking, pretty straightforward. They're esters formed by joining fatty acids with various types of alcohol. Within this category, the most recognizable examples are probably fats and oils, which we collectively call triacylglycerols, or TAGs for short.
Right, tags. And what's the actual difference between a fat and an oil in this context? Is it chemical?
No, it's purely physical, just the state at room temperature. Both are esters of fatty acids with glycerol. And oil is liquid at room temperature because its fatty acids are typically more unsaturated, which gives it a lower melting point.
Ah, the kinks we'll get to later.
Exactly. A fat is solid because it contains more saturated fatty acids, which allows it to pack together more tightly. Chemically, though, they are the same type of molecule.
Got it. Okay, beyond tags, we also have waxes in the simple lipid category. What are they?
Waxes are similar to tags in being esters, but they swap out glycerol for alcohols other than glycerol.
Yeah.
These are typically very long-chain alcohols, like cetyl alcohol is a common example.
Okay.
This structure makes waxes extremely hydrophobic and chemically stable. They function primarily as protective coatings. In nature, think of the waxy coating on a plant leaf or the sebum on animal skin. Industrially, they show up everywhere, from specialized lubricants and polishes to cosmetics. They're kind of nature's ultimate sealing agents.
Okay, so if simple lipids are about basic storage and sealing, the next category complex or compound lipids, this is where the architecture really starts to get interesting. What defines this group?
Complex lipids basically take a simple lipid structure and then attach additional groups. So we're talking about molecules that now incorporate non-lipid components like, say, phosphate or carbohydrates or proteins. This addition immediately gives them polarity and structural utility.
And the first, and you could argue maybe the most important group here, are the phospholipids.
Absolutely. Phospholipids are mission critical. They all contain phosphoric acid, which provides this highly polar, hydrophilic, water-loving head group. They're then subdivided based on their core alcohol backbone. The largest group are the glycerophospholipids, which use glycerol as the alcohol classic examples being lecithin, which is phosphatidylcholine, and cephalin phosphatidylethanolamine.
Okay, glycerol backbone. And what happens if that backbone changes? You mentioned another.
Right. If the alcohol changes from glycerol to sphingosine, which is a more complex long-chain amino alcohol, we get sphingospholipids. The most famous example here is probably sphingomyelin.
Sphingomyelin, okay.
And that shift from the flexible glycerol backbone to the more rigid sphingosine backbone often signals a shift toward more specialized, less fluid structural roles, particularly in nervous tissue.
That distinction, glycerol for maybe more fluidity, sphingosine for rigidity and structure, that's actually a really helpful way to remember that. Okay, that leads us directly then to the glycolipids.
Right. Glycolipids, often called glycosphingolipids, they maintain that rigid sphingosine backbone. They contain fatty acid, carbohydrate, and a nitrogenous base. What's really unique about them, and kind of a good way to remember the classification, is that they're defined by the absence of both glycerol and phosphate.
Ah, okay. No phosphate.
No phosphate. They are carbohydrate-containing lipids crucial for things like cell recognition and surface properties.
Yep.
Examples are cerebrosides and the more complex gangliosides.
And finally, to complete this complex group, we have lipoproteins. These sound like they bridge the gap between fat and the watery environment of the blood.
That's exactly what they do. Lipids are, as we said, largely insoluble in blood plasma, which is aqueous. So lipoproteins are these macromolecular complexes, kind of like a micellar taxi service, where proteins wrap around the hydrophobic lipid core. This allows them to be safely transported through the circulatory system, delivering cholesterol and tags wherever they're needed.
Okay, so we've got the builders. Then our sources classify derived lipids. These are products of hydrolysis, right? Like free fatty acids, glycerol, steroid hormones, fat-soluble vitamins.
Exactly. And miscellaneous lipids, which are just compounds that have lipid characteristics but don't quite fit the other boxes neatly, like carotenoids or squalene.
Right.
And just to close the loop on classification, if we sort of mentally stripped away all the charge, the phosphate, the carbohydrate, we're left with the neutral lipids. These are the uncharged species like mono, di, and triacylglycerols, as well as cholesterol and its cholesterol esters.
Excellent. Okay, we've established the geography of the lipid family, you could say. Now, let's drill down into the simplest functional components, the absolute building blocks. Fatty acids.
Fatty acids really are the fundamental currency of lipids. Chemically, they're carboxylic acids, characterized by a long, nonpolar hydrocarbon side chain. While they do exist freely in small amounts, they are overwhelmingly found in their esterified form, locked into those tags, phospholipids, and sphingolipids we just talked about.
Yeah, I find the commonalities in natural fatty acids quite striking, particularly the carbon count. Why do most natural fatty acids have an even number of carbons, usually what, between 14 and 20?
Yeah, that's a direct molecular signature of their biosynthesis. Our bodies assemble fatty acids by sequentially adding 2 carbon units derived from acetyl-CoA. It's like a metabolic conveyor belt that only deals in pairs.
Okay, that makes sense.
And this explains why palmitic acid, which is 16 carbons, and stearic acid, 18 carbons, are the most dominant examples found in nature. We classify them by chain length, too. Short, less than 6 carbons. Medium, 8 to 14. And long, 16 up to 24.
Beyond size, though, the most important classification seems to be based on saturation. This really dictates their shape and flexibility, right?
Absolutely. Saturation refers to the presence or absence of double bonds in that hydrocarbon chain. Saturated fatty acids have 0 double bonds. Their chains are straight and, well, relatively rigid. They're named with the suffix enoic.
Like octanoic acid.
Exactly. Unsaturated fatty acids have 1 or more double bonds, and this gives them chemical flexibility and, crucially, affects their shape. We differentiate between monounsaturated, or MUFA, just 1 double bond, and polyunsaturated PUFAs, 2 or more double bonds.
Okay, now let's tackle the shorthand notation. This is where the chemistry becomes almost like a concise language. How do we quickly describe the entire structure of a fatty acid?
Yeah, the standard notation is built on usually 3 numbers or pieces of information. It starts with the total number of carbons in the chain, followed by a colon, then the number of double bonds, and finally the positionness of the double bonds, counting from the carboxyl end, which is C1.
Okay, so using the examples from the source material. Stearic acid, which is saturated, 18 carbons. That's just 18 points.
Simple. And oleic acid, the main monounsaturated fatty acid in olive oil, has 18 carbons and 1 double bond. And that bond starts at position 9. So it's 18.19.
18.19.
The system is very precise. It tells you exactly where that single double bond is located relative to the carboxyl head group.
And then there's also the Greek numbering system, starting at C2, the alpha carbon, beta, gamma. But the system most people probably hear about is the omega system.
Yes, the omega carbon is the terminal methyl group, the absolute last carboxyl group. When we number from this terminal end, we establish omega $3, omega $6, omega $9. This system is used because, well, the body's metabolic machinery, when it adds carbons to a chain or modifies it, usually works from the carboxyl end. But when talking about essential status, the distance of the first double bond from that omega end is what's crucial. That's why you hear so much about omega-3 fatty acids in fish oil supplements, for example.
That brings us directly to the concept of essentiality. What makes linoleic acid and linolenic acid and linolenic acid and omega-3 essential fatty acids? Why can't we make them?
Right. Essential fatty acids, or EFAs, are PUFAs that our bodies cannot synthesize. We must get them from our diet. The biochemical reason is actually fascinating. Humans, like all mammals, lack the specific desaturase enzymes that are capable of introducing double bonds beyond the C9 and C10 positions, counting from the carboxyl end.
Great.
We can make an 18.1 fatty acid, like oleic acid, 18.19, but we simply cannot insert a double bond further down the chain, say at C12 or C15. Linoleic acid, 18.2, and linolenic acid, 18.3, contain those crucial double bonds at positions 12 and 15 respectively, making them biochemically essential for us.
So because we can't perform that one specific enzymatic step, we rely entirely on plants and microorganisms to do it for us. It's outsourcing, basically.
Pretty much, yes.
The source material also notes that arachidonic acid, another important PUFA, also becomes essential if its precursor, linoleic acid, is missing from the diet.
That is correct.
Right.
Arachidonic acid, which is 20.4, can be synthesized in the body, but only from linoleic acid. So if the dietary supply of the precursor, linoleic, is deficient, then arachidonic acid itself effectively becomes essential. You have to get it directly from food.
Yeah.
The whole metabolic pathway relies on that initial essential input.
And what are the physical consequences if the body is starved of these essential building blocks? What happens?
Well, the deficiency manifests quite visibly, actually, as a condition called phrinoderma, which literally means toad skin.
Toad skin.
Yeah, it's a harsh reminder that these molecules are structural, not just fuel. Phrinoderma is characterized by hyperkeratosis, meaning these horny, rough eruptions, particularly on the limbs, back, and buttocks. You also see thinning hair, and importantly, a major impairment of wound healing. It tells us that these PUFAs are absolutely necessary for maintaining the integrity and proper hydration of the skin barrier, really confirming their foundational role in cellular architecture.
Okay, now let's talk about shape, because the shape of the fatty acid is going to determine the structure of the wall it builds, right? We have to discuss isomerism, the famous kink in the chain.
Yes, this is one of the most critical structural concepts in biochemistry, especially for membranes. Unsaturated fatty acids exhibit geometric isomerism around the axis of the double bond. Basically, the way the groups attached to the double bonded carbons are positioned determines whether the molecule is straight or bent.
And the natural state, the one found most often in nature, is the cis configuration. Tell us about the kink.
Right. In the cis configuration, the hydrogen atoms, or the carbon groups attached to the double bond carbons, are oriented on the same side of the bond. And this isn't just a slight bend, it introduces a really pronounced structural dislocation, a molecular kink. Oleic acid, for example, is often described as being sort of L-shaped because of that bend.
L-shaped, okay.
And yet, over 99% of naturally occurring unsaturated fatty acids are found in this cis configuration.
In the opposite structure, the trans configuration, we hear a lot about trans fats.
In the trans configuration, the groups are on opposite sides of the double bond. This counteracts the bend, allowing the chain to remain relatively straight, much more similar in shape to a saturated fatty acid. Elatic acid is the trans isomer of oleic acid. While trans isomers are actually less stable thermodynamically, the critical importance of the cis-trans difference lies in how they pack together.
So why does that kink matter so much for the cell membrane?
Because the kinks caused by those cis bonds prevent the tight crystalline packing of the fatty acid tails within the membrane. Imagine trying to tightly stack a pile of L-shaped bricks versus straight rectangular bricks.
Right, you get gaps.
Exactly. The L-shaped bricks create gaps and maintain space. These gaps are absolutely essential. They ensure that the lipid bilayer maintains the necessary fluidity, a kind of semi-liquid state, which is crucial for things like protein movement within the membrane, signal transduction, and just ensuring the membrane doesn't freeze solid at body temperature. Trans fats lacking that kink can pack more tightly, potentially reducing membrane fluidity and making it more rigid.
That makes the molecular shape absolutely central to the viability of the cell. Okay, we examined the building blocks. Let's transition now to the biggest reservoirs. Triacylglycerols, or TAGs. These are the real champions of energy storage.
They truly are. TAGs are the most abundant lipid group in most organisms, and certainly the most energy-dense fuel reserve the body possesses. Structurally, they are nonpolar esters of glycerol linked to 3 fatty acids. Their complete lack of polarity means they are very hydrophobic and essentially entirely insoluble in water.
Let's quantify that storage capacity. The source material provides a really stunning statistic about the human reserve.
It is pretty stunning, yeah. The average fat reserve, about 20% of body weight for men, maybe 25% for women, is sufficient to meet the body's total caloric needs for 2 to 3 months.
2 to 3 months, wow.
Yeah. Now contrast that with carbohydrate storage, which is primarily glycogen in the liver and muscles. That's sufficient for maybe a single day of normal activity. The difference is huge.
Why is that difference so massive? Why are fats such a superior fuel source in terms of storage?
Well, there are two main reasons. First, the fatty acid tails are chemically far more reduced than glucose. This means their oxidation yields about twice the energy per gram, around 9 kilocalories per gram for fat, versus only about 4 kilocalories per gram for carbohydrates or protein.
Okay, more bang for your buck, chemically.
Exactly. Second, and perhaps more importantly for overall weight, tags are stored in an anhydrous state, meaning without associated water. Glycogen, by contrast, is polar and binds large amounts of water, about 2 grams of water per gram of glycogen. So, if we had to store that 2 to 3 months worth of energy purely as glycogen, we would simply collapse under the massive extra water weight we'd have to carry around. Lipids are just the most efficient, lightweight fuel package evolution could devise for long-term storage.
This massive fuel reserve is stored primarily in specialized cells, adipocytes, and adipose tissue. And critically, as our sources note, these tags are not typically found in cell membranes. Their role is strictly storage and insulation, right?
Precisely. They sit there, metabolically quite inert, until they're needed for fuel or perhaps for insulation against cold or as padding against mechanical shock. They're stored under the skin, the subcutaneous layer, and around the abdominal organs.
Our sources also highlight the need for something called stereospecific numbering, or the SEN system, when naming glycerol-based lipids. Why does a molecule that looks chemically symmetrical glycerol need this kind of specific 3D nomenclature?
Yeah, this is a perfect example of where simple chemistry on paper meets the reality of biology. While C1 and C3 of the glycerol molecule look identical if you just draw it flat, as soon as a substitution occurs at one of those positions, like adding a phosphate group to make a phospholipid, the central carbon, C2, becomes chiral.
Ah, okay, it gains handedness.
Exactly. It lacks a plane of symmetry. This means that C1 and C3 are no longer truly identical in a three-dimensional enzymatic context. Enzymes are highly specific and can absolutely distinguish between, say, synglycerol 1-phosphate and synglycerol 3-phosphate. So the thinged prefix ensures unambiguous naming that accurately reflects how the cell's machinery actually processes the molecule, recognizing its specific chirality.
That adds necessary context to what might seem like just extra jargon. Okay, moving on to the practical side. Understanding the properties of fats through some analytical tests, let's look at the breakdown mechanisms first: hydrolysis and saponification.
Okay, hydrolysis is the general term for the breakdown of the ester bonds linking fatty acids to glycerol. This is a crucial process during digestion, catalyzed by lipase enzymes, and also when the body needs to mobilize stored fat for energy from adipocytes. Saponification is more specific. It's the chemical hydrolysis using a strong alkali, like Li, sodium hydroxide, or potassium hydroxide, KOH. This breaks the ester bond to yield glycerol and the salts of the fatty acids, which are the basic chemical ingredients of soap. Hence the name saponification, meaning soap making.
Arcuona, the classic soap molecule. Okay. And then we get to rancidity, the enemy of food storage and pleasant-smelling kitchens.
Yes, rancidity is basically fat deterioration, leading to that unpleasant smell and taste. It's a major concern for the food industry, obviously. We generally see two main types. First is hydrolytic rancidity. This happens when microbes, like bacteria, produce lipases that partially break down the tags, releasing free fatty acids. If these are short-chain fatty acids, like butyric acid from butterfat, they're volatile and smell pretty terrible.
Okay, that's bacterial action. What's the other type?
The other, often more problematic type is oxidative rancidity. This is where atmospheric oxygen directly attacks the double bonds in unsaturated fatty acids. This process cleaves the chains and forms highly reactive and smelly compounds like aldehydes and ketones.
Which means highly unsaturated fats, like those in fish oil or maybe flaxseed oil, are much more susceptible to going bad quickly than, say, lard or coconut oil, which are more saturated.
Absolutely. The more total bonds, the more sites for oxygen to attack. And that's why we need defenses against it. Commercial fats often rely on synthetic antioxidants like BHA and BHT, or natural ones like tocopherols, which is vitamin E, to basically quench those initiating free radicals before they can start the chain reaction of oxidative rancidity.
And our sources also draw a parallel to something similar happening inside us, in vivo lipid peroxidation.
Exactly. The same kind of free radical chemistry can attack lipids within our own bodies, particularly in cell membranes. This can damage tissues and is implicated in processes like aging and various chronic diseases. Fortunately, the body has its own defenses, including natural antioxidants like vitamin E and enzymes like superoxide dismutase to combat this ongoing oxidative stress.
Okay, let's discuss the three specific purity tests outlined in our sources. These seem quite clever ways to chemically fingerprint fats, starting with the iodine number.
Yeah, the iodine number is defined as the grams of iodine absorbed by 100 grams of fat. Iodine reacts specifically and quantitatively with carbon double bonds. Therefore, the iodine number is directly proportional to the degree of unsaturation in the fat sample. It's a quick and elegant way to assess how many double bonds are present and thus how liquid or unsaturated a fat is likely to be.
And the ranges given are huge, almost unbelievable. Linseed oil, which is highly polyunsaturated, has an iodine number way up at 175, 200, while highly saturated coconut oil is down at only 710. It really is a chemical fingerprint.
It is. Then we have the saponification number. This is defined as the milligrams of potassium hydroxide, KOH, required to completely hydrolyze or saponify 1 gram of fat. This test essentially measures the average molecular size of the fatty acids in the tags.
How does it measure size?
Well, think about it. If the fatty acids are short, it takes more molecules of tag to make up 1 gram of fat. Since each ester bond needs 1 molecule of KOH to break it, a fat composed of shorter chain fatty acids will require more KOH per gram. So a higher saponification number indicates the presence of shorter chain fatty acids on average.
Ah, okay. More molecules per gram means more KOH needed. And coconut oil, which we know has lots of medium chains, has a very high value given, 250, 260.
Exactly. And finally, there's a test that's quite specific, almost like a forensic test for one very important dairy product, the Reichert-Meisel RM number.
What's this one for?
The RM number is a classic test used almost exclusively to check for the purity of butter. It specifically quantifies the amount of soluble, volatile fatty acids, primarily butyric acid and caproic acid, that can be distilled from the saponified sample. Since these short volatile acids are characteristic of butterfat and virtually absent in most other common fats and vegetable oils used as adulterants.
Like margarine or something added to bulk it out.
Precisely. Butter has a characteristic RM number, typically between 25 and 30. Any other oil used to adulterate the butter will yield a number less than 1, usually. It's an incredibly precise measure of authenticity for butter.
That wraps up the fuel and testing aspect quite nicely. Now let's pivot entirely to section 4 in our sources. Structural and regulatory lipids. These are the molecules whose primary job isn't energy storage, but rather architecture and signaling. We begin with the big ones, phospholipids, the membrane scaffolding.
Right. Phospholipids are absolutely the central component of all biological cell membranes, forming that essential bilayer structure. We noted the 2 main subclasses earlier based on the alcohol backbone, glycerophospholipids and sphingophospholipids. Let's dive into the specific roles of the major glycerol-based ones first.
Okay, we have lecithin, which is properly called phosphatidylcholine. The sources say it's the most abundant. What makes it so mission critical?
Lecithin serves as a general structural component in many membranes, and it's also a storage source for choline, which is an essential nutrient involved in neurotransmitter synthesis and other things. But its most dramatic and honestly life-saving role lies in a specific derivative, dipalmitoyl lecithin.
Dipalmitoyl lecithin, what does that do?
This specific molecule is the primary constituent of lung surfactant. Its main job is to dramatically lower the surface tension inside the tiny air sacs in our lungs, the alveoli. Without sufficient dipalmitoyl lecithin, the high surface tension of the water lining the alveoli would cause them to collapse completely when the bee exhales.
Wow.
Its absence or insufficient production is the direct cause of respiratory distress syndrome, RDS, in premature infants, whose lungs haven't matured enough to produce it adequately. It really highlights how a single specific lipid molecule can be the difference between life and death outside the womb.
That is an incredible example of molecular specialization. What about cephalins and phosphatidylinositol PI? They're also mentioned.
Cephalins, which are phosphatidylethanolamine, are structurally similar to lecithin, but use ethanolamine instead of choline as the head group. They are particularly important in nervous tissue and play roles in processes like blood cytosin or vasopressin, bind to receptors on the cell surface. Enzymes are activated that immediately cleave PI, which is sitting right there in the inner leaflet of the membrane. This cleavage releases secondary messengers into the cell, cytoplasm like inositol triphosphate IP3 and diacylglycerol, DAG.
So PI is essentially a localized reservoir of chemical command waiting for the signal.
Exactly. It's perfectly positioned. When the cell gets that external message, PI provides the internal messengers that then initiate a cascade of events inside the cell, perhaps leading to the release of stored calcium, which then triggers a specific cellular response. It effectively mediates the hormonal action. It's like the molecular switchboard right at the membrane.
Fascinating. And the last glycerophospholipid mentioned is the complex structure of cardiolipin.
Yeah, cardiolipin is structurally unique because it's essentially a double phospholipid. It consists of two phosphatidic acid units joined together by an extra glycerol molecule in the middle. Its location is also highly specific. It is found almost exclusively in the inner mitochondrial membrane.
The powerhouse of the cell.
Exactly. It's essential there for organizing the protein complexes involved in the electron transport chain and ATP synthesis, basically powering the cell's energy generation. Its decreased levels or altered structure are directly linked to severe inherited mitochondrial disorders, such as Barth syndrome, which affects the heart and muscles.
Okay, now we switch backbones, away from glycerol to the sphingophospholipids featuring sphingomyelin.
Right. Sphingomyelins, using the aminoalcohol sphingosine instead of glycerol as their backbone, are particularly crucial in nervous tissues. They're major components of myelin, that fatty sheath that insulates nerve axons.
Ah, the insulation for nerve impulses.
Precisely. Myelin allows electrical signals to travel much more rapidly and efficiently along the nerve. Without proper myelin, nerve transmission slows dramatically, which is what happens in diseases like multiple sclerosis. Now, the core structural unit of all sphingolipids, including sphingomyelin, is ceramide. This is a sphingosine molecule linked via an amide bond to a fatty acid.
And ceramide itself is one of those molecules with a dual personality, isn't it? It's structural, but also a signal.
Absolutely. Structurally, specific ceramides, particularly those containing ultra-long chain fatty acids, up to 30 carbons long, are major components of the outermost layer of the skin, the stratum corneum. They are critical for regulating water permeability and maintaining skin hydration, literally sealing us from the outside world.
Okay, skin barrier. What's the signaling role?
Internally, ceramide is a potent secondary messenger molecule. It's involved in regulating crucial cellular decisions, including cell differentiation, proliferation, and most significantly, programmed cell death or apoptosis.
Programmed cell death, so it can act like a chemical executioner when needed.
In a sense, yes. It can trigger the cascade of events that lead to the cell dismantling itself in a controlled way. So it has this profound dual role, maintaining the barrier of life on the outside and signaling for its end on the inside when necessary.
That gives the sphingolipids a massive structural and regulatory footprint.
Okay, moving to the related glycolipids, which our sources say serve as cell identifiers.
Yes, glycolipids, which also use sphingosine as the backbone, so they're often called glycosphingolipids, are essential components of cell surfaces, particularly on the outer leaflet of the plasma membrane, and are also abundant in nervous tissues. The simplest are the cerebrosides.
Cerebrosides.
These contain a ceramide molecule linked to a single sugar, usually either glucose or galactose. For example, galactocerebroside, which is found predominantly in brain tissue, contains galactose and often the specific fatty acid cerebronic acid. They act as recognition sites on the cell surface, important for cell-cell interactions.
The sources also make an interesting side note here about cholmogric acid. Ah, yes, an interesting historical detail. Cholmogric acid is an unusual cyclic fatty acid found in cholmugra oil. This oil was historically, for centuries actually, one of the few treatments available for leprosy, although its mechanism wasn't understood and efficacy was limited. It's just an interesting example of an unusual lipid structure with a historical medical connection.
Okay. And the most complex glycolipids are the gangliosides.
Yes, gangliosides are the most complex glycosphingolipids structurally. They're heavily branched and contain one or more molecules of N-acetylnuraminic acid, which is a sialic acid, often abbreviated as NANA. This gives them a net negative charge.
And where are they found?
They're found predominantly in the nervous system, particularly concentrated in the ganglions of the brain, hence the name. Their complex carbohydrate structures are crucial for nerve transmission and cell-to-cell communication, acting as receptors or modulators. Their importance is starkly highlighted by certain genetic diseases.
Like Tay-Sachs.
Exactly. Tay-Sachs disease involves the pathological accumulation of a specific ganglioside called GM2 within the lysosomes of nerve cells, because an enzyme needed to break it down is missing. This accumulation leads to severe neurodegeneration and is ultimately fatal. It shows how critical the normal metabolism of these complex lipids is.
Okay, let's pivot now to the final major class of structural and regulatory lipids discussed, steroids. These look completely different from the long, often floppy fatty acid chains we've been discussing.
They really do. Steroids are defined by a fundamentally different structure, a rigid fused ring system. This core structure is called the cyclopentanohydrophenanthrene, CPPP, nucleus.
Catchy name.
Ah, yes. It's composed of 3 fused 6-carbon rings, labeled A, B, and C, which form the phenanthrene part, fused to a single 5-carbon ring, the D ring or cyclopentane part. This multi-ring structure makes them relatively rigid and flat molecules, the structural antithesis of the flexible fatty acid tail. If a steroid molecule contains one or more hydroxyl H groups, we classify it specifically as a sterol.
And the most famous, most abundant animal sterol found in pretty much every single one of our cell membranes is cholesterol.
Cholesterol is absolutely non-negotiable for animal life. Structurally, it has that core CVPP ring system with a single hydroxyl group at position C3. This gives it a tiny bit of polarity, making it weakly amphipathic. It also has a double bond between C5 and C6 in the B ring, and an 8-carbon branched hydrocarbon side chain attached at C17. It's synthesized mainly in the liver, but present in all tissues, transported via those lipoproteins we mentioned earlier.
Given its often negative reputation in popular health discussions, it's easy to forget that cholesterol performs absolutely vital functions.
Oh, its functions are myriad and essential. Within the cell membrane, its rigid planar structure inserts between the phospholipid tails. There, it helps maintain structural integrity, and importantly, it modulates membrane fluidity, acting as a kind of fluidity buffer, preventing the membrane from becoming too fluid in high temperatures or too rigid at low temperatures.
Okay, membrane structure. What else?
In nervous tissue, it's a major component of myelin, acting as an insulating cover, crucial for the speed and efficiency of electrical impulse transmission. It's a poor conductor of both heat and electricity. But perhaps most importantly, cholesterol is the essential precursor molecule for the synthesis of a whole host of other critical regulatory molecules.
Like what?
Like all the bile acids needed for fat digestion, all the adrenal cortical hormones such as cortisol, regulating stress response and metabolism, and aldosterone, regulating salt balance, all the sex hormones, testosterone, estrogen, progesterone, and vitamin D. Without cholesterol as the starting material, the body simply cannot make any of these life-sustaining compounds.
So indispensable. And our source briefly mentions ergosterol in the context of fungi sterols.
Right. Ergosterol is the principal sterol found in the membranes of yeast and fungi, essentially serving the same structural role in their membranes that cholesterol serves in ours. It's biochemically relevant to us mainly because when yeast or fungi containing ergosterol are exposed to ultraviolet UV light, the B ring of the ergosterol molecule can be opened up, transforming it into a compound that possesses vitamin D activity specifically, vitamin D2 or ergocalciferol.
Ah, the source of vitamin D in irradiated mushrooms, for example.
Exactly.
Okay, we've covered structure, fuel, and signaling in detail. Now let's bring it all back together in the final section by focusing on the defining characteristic that allows these structural lipids to actually build the cell wall and other structures, the amphipathic nature.
Yes, the amphipathic nature is really the crucial physical property underpinning membrane structure and lipid transport. Amphipathic, as the name suggests, amphi meaning both, pathic meaning loving, describes a molecule that possesses both a hydrophobic, water-repelling nonpolar region and a hydrophilic, water-loving polar region within the same molecule.
The dual personality molecules.
Precisely. We've seen many examples.
Yeah.
Free fatty acids, with their carboxyl head and hydrocarbon tail, phospholipids, polar head group, non-polar tails, single lipids, bile salts, and even cholesterol, which is weakly amphipathic due to that single hydroxyl group.
The phospholipid structure is probably the best visual example. That distinct polar head and the long nonpolar tail. So what happens when you dump these dual citizens into water? They can't fully dissolve, but they can't fully avoid it either.
Right. They spontaneously self-assemble into structures that minimize the unfavorable contact between their hydrophobic tails and the surrounding aqueous environment, while maximizing the contact between their hydrophilic heads and water. This natural tendency, driven purely by thermodynamics, dictates the formation of two major types of structures, depending on factors like the lipid's shape and concentration.
Okay, first structure, the micelle.
In a micelle, the amphipathic lipids arrange themselves into a spherical aggregate. All the polar heads face outward, happily interacting with the surrounding water, while all the nonpolar tails tuck inward, pointing towards the center, forming a tightly packed, water-free hydrophobic core.
And this is important for digestion.
Absolutely essential. Micelle formation, which is highly facilitated by bile salts, which are themselves amphipathic in the gut, is crucial for solubilizing dietary lipids and the products of their digestion, allowing them to be absorbed across the intestinal wall into the body. It effectively allows fats to move through an aqueous environment.
Okay. And the second structure, the one that's literally the foundation of every cell on Earth, the lipid bilayer.
The bilayer is the fundamental structural realization of the anthropathic nature in a biological context. Here, lipids, primarily phospholipids and glycolipids, spontaneously arrange themselves into a double layer. The hydrophobic tails of both layers face inward towards each other, creating a continuous, non-polar core in the middle of the membrane. Meanwhile, the hydrophilic heads of both layers face outward, one layer facing the external aqueous environment outside the cell, and the other layer facing the internal aqueous cytoplasm inside the cell.
Forming a barrier.
Exactly. This bilayer formation is thermodynamically stable, acts as a selectively permeable barrier, and forms the basic structural framework of every single cell membrane and the membranes of internal organelles like the nucleus or mitochondria.
It is truly remarkable that the boundary of life itself is essentially maintained simply by the physics of how water interacts with these dual personality molecules. Our source also points out a modern technological application of this self-assembly principle, liposomes.
Yes, liposomes are basically synthetic, microscopic spherical vesicles created in the lab, usually by subjecting amphipathic lipids like phospholipids to sonification using sound waves or other dispersion methods in water. They are essentially artificial lipid bilayers enclosing a small aqueous core.
And why are they useful?
This technology has become incredibly important in modern medicine and research, primarily for drug delivery. Liposomes can be engineered to encapsulate drugs. Either hydrophobic drugs can be tucked into the lipid bilayer itself, or hydrophilic drugs can be carried within the aqueous core.
Like a tiny delivery vehicle.
Precisely. This allows for targeted delivery of the drug to specific tissues, can protect the drug from degradation in the bloodstream, potentially reduce side effects, and control the release rate. It's a direct application of understanding lipid self-assembly.
We've completed a massive circuit today, haven't we? Starting with the very definition of lipids by their hydrophobicity, their hatred of water. We classified them into simple storage fuels like the tags, the complex structural components like phospholipids and glycolipids, and the rigid regulators like steroids. We detailed the crucial structural importance of that fatty acid kink and the sort of elegant quantitative power of those chemical purity tests.
And we close by circling back to the fundamental principle. The ability of amphipathic lipids to spontaneously create a sealed yet dynamic boundary, the lipid bilayer, is arguably the fundamental physical act required for life as we know it. There's so much more than just fats. They are the architects that literally separate self from non-self, defining the very limits and compartments of the cellular world. We also touched on their integrated roles in membranes as precursors for fat-soluble vitamins and hormones, and in metabolic regulation. And of course, the implications when lipid balance goes wrong, leading to disorders like obesity, atherosclerosis, and diabetes mellitus.
Okay, to leave you, our listeners, with a final thought that really connects structure, signaling, and even cellular fate in a fascinating way. We learned earlier that derived lipids, specifically the sphingolipid component ceramide, can act as powerful secondary messengers inside the cell. And one of its key roles is regulating critical processes like programmed cell death or apoptosis. Now think about this profound connection for a moment. The essential boundary of the cell, the membrane itself, is made primarily of phospholipids and sphingolipids, like sphingomyelin, which contains ceramide. Derived from these very boundary components are molecules, like free ceramide, which can then determine the ultimate fate of that cell. So the very components that define the boundary of life, ensuring structural stability and controlling things like skin water permeability, are also intrinsically linked to issuing the ultimate chemical command for that life to end, regulating everything from hydration to final cellular apoptosis.
It really suggests that the boundary of life intrinsically holds the command code for its own termination when necessary. It's a truly compelling integration of structure,
Roadmap for these amazing structures. It takes you right from the tiniest chemical bits all the way up to their final functional 3D shapes.
it really covers the ground.
It does. So our mission today is really to give you a bit of a shortcut, right? A high-level view of exactly how these molecules, which, let's be clear, form the basis of structure and function in pretty much every living system, how they're put together, how they stay stable, and ultimately how they behave.
And the scale is just immense when you look at it.
Oh, totally. I mean, the raw statistics are staggering. Proteins are easily the most abundant organic molecules in any living system. They make up something like 50% of the cellular dry weight. That's huge.
Half the weight, when you take out the water, it tells you how central they are.
And they're needed everywhere. The sources break down their roles into two really broad categories. First, you've got the static or structural roles. This is the physical stuff.
The scaffolding.
Exactly, the scaffolding. Think of it like the brick and mortar of the body. You've got collagen in your tendons, your skin, keratin making up hair and nails. It's all about providing strength and form.
Solid, dependable structures, but then you have the other side of the coin.
Right, the dynamic roles. And arguably, these are maybe the most crucial for just keeping things running day to day, wouldn't you say?
Well, absolutely. These are the functional heavy lifters.
Yeah.
The real working horses inside the cell. They're acting as enzymes, speeding up basically every essential chemical reaction.
Millions of times faster sometimes.
Unbelievably faster. They're hormones carrying vital signals all over the body. Immunoglobulins for defense against invaders. They're key players in muscle contraction, genetic control. It's the cell's operational staff, really.
And the efficiency of life, it just boils down to how precisely these proteins function.
It does.
And here's what I find truly elegant. Every single one of these massive, complex, multitasking structures, whether it's that structural collagen or a super specific catalytic enzyme, is built from the exact same simple foundation.
20 building blocks.
Right. They are all polymers, these long chains built exclusively from alpha amino acids. So if the finished protein is like some kind of sophisticated, custom-designed building...
And then the amino acids are the essential bricks. Chemically versatile bricks, mind you.
Exactly. Versatile bricks.
So to really get into this deep dive, we absolutely have to start with the brick itself. Understand the amino acid first. Okay, so let's look at the core structure of an amino acid. Architecturally, it's simple but, well, profound in its implications. Everything centers around one specific carbon atom.
The alpha carbon, right, the alpha carbon.
That's the one. The alpha carbon, it's the central anchor. And it has to be attached to 4 different things, typically. You've got the basic amino group that's made from an H2X, and the acidic carboxyl group made from COACs.
Okay, basic and acidic. And critically, there's the side chain. That's the R group, isn't it?
Exactly, the R group. And this is the game changer. It's the identifier, the chemical signature that dictates pretty much everything about that specific amino acid. Its size, its charge, whether it likes water or hates it.
It's personality almost.
It's chemical personality, absolutely. And it dictates where it's likely to end up when the final protein folds up. It's what makes each of the 20 standard amino acids unique. Hashtag 1.1, the general structure and ionization. And they're called alpha amino acids specifically because both of those defining groups, the amino and the carboxyl, are bound to that central alpha carbon. But here's where it gets really interesting. How they actually exist in a biological system, like in our cells.
Right, because they're not usually just floating around neutral, are they?
Almost never. Not in that simple textbook neutral form. In the biological system, especially around physiological manthrem, which is near neutral, well, as an acidic carboxyl group readily gives up a proton. It becomes negatively charged.
Okay. Loses a proton.
And the basic amino group, it picks one up, it becomes positively charged, methylamine H3 plus CO. So the resulting molecule is what we call a zwitterion, or dipolar ions.
Zwitterion, sounds fancy. So it's got both a positive and a negative charge at the same time.
Exactly, simultaneously. Which means even though it has charges, the molecule overall is electrically neutral. And this perfect balance happens only at a very specific pH value for each amino acid.
Which is its isoelectric pH, or PI.
Precisely, it's PI.
Oh.
And this dual nature, this ability to carry charge but be neutral overall, is why amino acids are called ampholytes. They can act as both weak acids and weak bases.
Ah, so that's key for buffering, right? Resisting pH changes.
Absolutely fundamental for their buffering capacity and how they interact in solution. It's critical.
Okay, and this structure also relates to their shape in 3D.
It does, directly. Remember, the alpha carbon is bound to 4 different groups. You've got the amino group, the carboxyl group, a hydrogen atom, and that unique R group.
Unless it's glycine.
Good point. Glycine is the exception. Its R group is just another hydrogen atom. So the alpha carbon in glycine is attached to only 3 different types of groups. But for all the others,
19 others.
The other 19, the alpha carbon is bound to 4 distinct groups. That makes the alpha carbon asymmetric, or chiral. And this asymmetry leads to what we call optical isomerism. They can exist in 2 mirror image forms.
L form and D form.
L and D isomers. And when it comes to life as we know it, our bodies are incredibly selective. Proteins in basically all living systems are made up exclusively of the alpha amino acids.
Only the L form. What about the D form? Does it just not exist?
Oh, D amino acids definitely exist in nature, but they're usually found in more specialized places, like in the cell walls of certain bacteria or in some antibiotics produced by microbes. But for the core machinery of life, the proteins doing all the work, It's strictly L amino acids. It highlights this really strict chiral purity that life seems to require. Tag 1.2 structural classification. The 7 families.
Okay, so we've got these 20 versatile bricks. Now, to make sense of how a long chain of them might fold up, we need to organize them somehow. The source material groups them based on the chemical nature of that R group, right? Into 7 families.
Yes, 7 distinct structural categories. It's a useful way to think about them. We can start with the simplest, the ones with aliphatic side chains.
Aliphatic meaning just carbon and hydrogen, basically. Non-aromatic.
Pretty much. Simple hydrocarbon-based groups.
Yep.
This includes the smallest ones, like glycine and alanine. And it also includes the important branched chain amino acids, or BCAAs.
Oh, the BCAAs. Valine, leucine, isoleucine, you hear about those in supplements?
You do. They're highly hydrophobic water hating, and they play really critical roles, especially in things like muscle metabolism and cell signaling.
Okay, family 1, aliphatic. What's next?
Next, we have amino acids containing an oxygen group, specifically a hydroxyl group. These are the hydroxyl group containing amino acids. That's serine and threonine.
Serine and threonine. And that hydroxyl group, the Mothram A's, that makes them polar.
Highly polar, yes. Excellent sites for forming hydrogen bonds, which are crucial for structure. And importantly, they're key sites for post-translational modifications. Things like phosphorylation, adding a phosphate group, often happen right there on that hydroxyl group.
Which can act like an on-off switch for the protein's activity.
Exactly, a major regulatory mechanism. Okay, third family, the sulfur-containing amino acids. 2 main players here.
Cysteine and methionine.
Right. Cysteine is unique because it has a reactive sulfhydryl group. Now, this group can covalently bond with the sulfhydryl group of another cysteine molecule.
Forming cysteine.
Forming cysteine, yes, via a disulfide bond. This is a very strong covalent bond, and it's one of the most powerful forces for locking a protein's 3D structure into place.
Okay, so disulfide bonds are like structural staples. What about methionine?
Methionine contains a thioether group, methamet CH3, instead of the sulfhydryl. It's less reactive in that sense, but it's critically important because it almost always serves as the starting amino acid when a protein is being synthesized in the cell.
The start signal for translation. Got it. Sulfur done. What's family 4?
Moving on to the ones that carry a charge at neutral pH. First, the acidic amino acids and their amides. The acidic ones are aspartic acid and glutamic acid.
Sometimes called aspartate and glutamate?
Yes, because at physiological pH, they've lost a proton from their second carboxyl group in the R chain, so they carry a net negative charge.
Okay, acidic means negative charge, and they're amides.
Their amide derivatives are asparagine, from aspartic acid, and glutamine, from glutamic acid. The extra carboxyl group is converted to an amide group, methylmnh2y. This neutralizes the negative charge, but they remain very polar molecules, good for hydrogen bonding.
Okay, acidic and amides. Next must be
The counterparts, the basic amino acids. These are highly basic, meaning they readily accept protons, so they carry a net positive charge in the cell.
So acidic or negative, basic or positive. Who's in this group?
This group includes lysine, which has a long side chain ending in an epsilon amino group. Then there's arginine, which boasts the guanidino group that's a very stable, highly basic group. And finally, histidine, with its imidazole ring.
Histidine's interesting, isn't it? Because its charge can change around physiological pH.
It is. Its pKa is close to neutral, making it really versatile in enzyme active sites, acting as a proton donor or acceptor. But generally, these positive and negative R groups are essential for forming those ionic interactions, those salt bridges, that help stabilize the final 3D architecture.
Like little magnets holding parts of the chain together. Okay, 2 families left.
We then hit the ones with ring structures.
Yeah.
The aromatic amino acids. These are phenylalanine, tyrosine, and tryptophan.
Big, bulky rings.
Yes, and quite hydrophobic generally. They all contain these stable ring structures. Phenylalanine has a simple benzene ring. Tyrosine is similar, but with an added hydroxyl group, making it a bit more polar. And tryptophan has a complex double ring structure called an indole ring.
And these are the ones that absorb UV light, right? That's how we measure protein concentration in the lab.
Exactly. Their aromatic wings strongly absorb UV light around 280 nanometers. Very useful property. Okay, last one.
The odd one out.
The structural oddity, yes, the amino acid, proline. It's unique. Its side chain isn't just hanging off. It actually loops back and bonds covalently to the backbone nitrogen atom itself.
Forming that rigid ring.
A rigid pyrrolidine ring, yes. This means that instead of a standard primary amino group, Mathram NH2 like all the others, proline has a secondary imino group. And that rigid ring structure severely restricts rotation around the peptide bond it forms.
So it acts like a kink in the chain.
Precisely. It often introduces sharp turns or kinks in the polypeptide backbone. It can't fit easily into regular structures like the alpha helix, so it often acts as a helix breaker or a turn inducer. Very important for dictating the final fold. So, now that we have a feel for the structures, we can classify them in more functional ways. And probably the most important classification for predicting how a protein will fold is based on polarity.
Right, because proteins usually fold up in water inside the cell, so how the amino acids interact with water is key.
Absolutely key. Polarity dictates where an amino acid is likely to end up in the final 3D structure. So first group, nonpolar hydrophobic.
Water-hating.
Water-hating, no charge. This includes alanine, leucine, isoleucine, valine, methionine, phenylalanine, tryptophan, and also proline. Because they dislike water, they tend to aggressively cluster together in the interior of the protein.
To hide from the water, like an oil droplet forming.
Exactly. This hydrophobic collapse is often considered the primary driving force behind protein folding. Get the oily bits away from the water.
Okay, hydrophobic core. Then what?
Then we have the polar no-charge amino acids. This includes glycine, serine, threonine, cysteine, glutamine, asparagine, and tyrosine. They have groups like hydroxyls, the sulfhydryl, or amide groups. These don't carry a net charge at neutral pH, but they are polar.
Meaning they like water?
They're hydrophilic, yes. They can form hydrogen bonds with water and also with each other. So you tend to find these on the surface of the protein, interacting with the cellular environment, or involved in forming the hydrogen bonds that stabilize secondary structures.
Okay, non-polar inside, polar outside are bonding. And the charged ones.
Right, the charged ones. We have polar positive R group. These are the basic amino acids, lysine, arginine, and histidine. And polar negative R group. These are the acidic amino acids, aspartic acid and glutamic acid.
The bases and acids.
Exactly. These are usually found on the protein surface too, interacting with water or forming those crucial ionic interactions, the salt bridges with each other, which help lock the tertiary structure into its final shape. They also define the protein's overall net charge at any given pH.
Makes sense. Polarity drives folding. What about other ways to classify them?
Another really important classification, especially from a health perspective, is based on nutritional requirement. Our bodies are amazing chemical factories, but they can't synthesize everything from scratch.
Some building blocks we just have to eat.
Exactly. These are the essential or indispensable amino acids. For humans, there are generally considered to be 10 that are needed for proper growth and maintenance, and these must come from our diet.
Okay, 10 essential ones. Which are they?
The source lists the core 8 as valine-leucine, isoleucine, the BCAAs again, lysine, methionine, phenylalanine, threonine, and tryptophan. But we usually also include arginine and histidine in that essential list, particularly for certain life stages.
Why only sometimes?
They're often called semi-essential. Healthy adults can typically synthesize enough arginine and histidine for their needs. However, growing children or individuals under certain metabolic stress or illness might not make enough, so for them, a dietary supply becomes essential.
Gotcha. So essential and semi-essential. The rest we can make.
The remaining 10 are non-essential or dispensable. This means our bodies have the metabolic pathways to synthesize them, usually from other common metabolites. Examples include glycine, alanine, serine, proline, glutamate.
Okay. Essential, non-essential. Diet matters. Is there one more classification?
Yes, one more functional view, based on their metabolic fate. When a body breaks down amino acids, what happens to their carbon skeletons? Where do those carbons end up?
After the nitrogen is removed for urea or other things.
Precisely. Some amino acids are glycogenic. This means their carbon skeletons can be converted into intermediates like pyruvate or oxaloacetate, which can then be used to synthesize glucose or glycogen through gluconeogenesis.
So they can become sugar, like alanine, methionine.
Many of them, yes.
Yeah.
Others are ketogenic. Their breakdown products like acetyl-CoA or acetoacetyl-CoA lead directly towards the synthesis of fatty acids or ketone bodies.
They can become fat or ketones. Who are the purely ketogenic ones?
Only 2 are exclusively ketogenic, leucine and lysine, all else.
Leucine and lysine. Only fat ketones. Are there any that do both?
Yes, there are 4 clever ones whose carbon skeletons can enter both pathways. They are both glycogenic and ketogenic. These are isoleucine, phenylalanine, tryptophan, and tyrosine.
The aromatics plus isoleucine, so they offer metabolic flexibility.
Incredible flexibility. Their carbons can be shunted towards sugar production or fat ketone production depending on the body's needs at the time. It really highlights how interconnected these metabolic pathways are. Hashtag tag 1.4 non-standard amino acids beyond the 20.
Okay, this is where, as the source says, it gets really interesting. Because biology, well, it doesn't always stick neatly to that list of 20 standard amino acids encoded by the main genetic code, does it?
Not at all. The system has evolved some fascinating exceptions and additions. We absolutely have to talk about the unique standard amino acids first.
Like the 21st and 22nd?
Exactly. Selenocysteine is often called the 21st amino acid. As the name suggests, it contains a selenium atom instead of the sulfur atom found in cysteine. It gets incorporated into a special class of proteins called selin proteins, which are often involved in antioxidant defense.
But how does it get put in? I thought there were only codons for 20.
That's what's so amazing. The genetic code used for selenocysteine is actually UGA. Now, UGA is normally a stop codon. It tells the ribosome to terminate protein synthesis.
So how does it insert an amino acid instead of stopping?
The cell has evolved a complex machinery involving specific RNA structures and protein factors that, under the right conditions, essentially overrides the stop signal at that UGA codon and directs the ribosome to insert selenocysteine instead. It's a remarkable feat of genetic recoding.
Wow, repurposing a stop sign. And the 22nd?
The 22nd is pyrrolysine. This one is found mainly in certain methane-producing archaea and some bacteria. It uses the UAG stop codon, another stop signal, for its incorporation. Again, specialized machinery allows the cell to read UAG as insert pyrrolysine in the context of specific messenger RNAs.
So the cell is literally redefining its own genetic alphabet on the fly. That's incredible.
It really showcases the evolutionary plasticity of the genetic code. But beyond these genetically encoded exceptions, there are other non-standard amino acids crucial for biology.
Like ones that are modified after the protein is made?
Precisely. Derivatives in proteins, created by post-translational modification. These modifications often add crucial stability or function that the standard 20 can't provide alone.
The big examples are in collagen, right? That structural protein.
Absolutely. 4-hydroxyproline and 5-hydroxylysine. Normal proline and lysine residues within the newly synthesized collagen chains get hydroxylated, and a mathrm group is added.
And why is that important?
These hydroxyl groups are essential for forming the extra hydrogen bonds that stabilize the incredibly strong triple helix structure of collagen.
Right.
Without these modifications, the collagen molecule is weak and unstable.
Which is what happens in scurvy.
Exactly. Vitamin C is a required cofactor for the enzymes that perform this hydroxylation. No vitamin C, no stable collagen. That's why scurvy leads to bleeding gums. Core wound healing tissues literally start to fall apart.
A direct link from a vitamin to protein structure. Wow. Any other key derivatives?
Another important one is gamma-carboxyglutamic acid, cool Anatolica. This is found in several plasma proteins involved in blood clotting, like prothrombin. Specific glutamic acid residues get an extra carboxyl group added.
And that extra negative charge does what?
It allows the protein to bind calcium ions, mathrm Ka2 plus Vbit, very effectively. This calcium binding is absolutely critical for anchoring these clotting factors to phospholipid surfaces at the site of injury, initiating the coagulation cascade. Some blood thinners, like warfarin, actually work by inhibiting the enzyme that makes GLALA residues.
Fascinating. Okay, so we have modified amino acids within proteins. What about amino acids that aren't in proteins at all?
Yes, the non-protein amino acids. These have the basic amino acid structure, but are never incorporated into polypeptide chains during translation. Instead, they function as important molecules in their own right.
Like intermediates in metabolic pathways.
Exactly. The source mentions ornithine and citrulline. These are key intermediates in the urea cycle, the pathway our body uses to safely dispose of toxic ammonia by converting it into urea. They cycle through the pathway but never end up in a finished protein.
Okay. Any others?
Then you have amino acid derivatives that act as hormones or neurotransmitters. Thyroxine, the main thyroid hormone that regulates metabolism, is synthesized from tyrosine.
And the classic example must be GABA.
Gamma-aminobutyric acid, GABA, yes. It's the primary inhibitory neurotransmitter in the brain, crucial for calming nerve activity. And it's synthesized directly from glutamic acid, one of the standard 20. So these examples really show that the basic amino acid structure is incredibly versatile, used for much more than just building proteins.
All right, so we have a solid understanding of the bricks now, these versatile amino acids. Let's look at how we actually lay the foundation for a protein, which brings us to the primary structure.
Primary structure. This is simply the specific linear sequence of those alpha amino acids as they're linked together to form the polypeptide backbone. It's the fundamental blueprint.
And when we say blueprint, we mean it's absolutely critical, right? This sequence dictates everything else.
It is non-negotiable. This unique sequence is determined directly by the organism's genes, encoded in the DNA. This linear order which amino acid follows, which dictates every single subsequent step of folding. It determines the protein's final 3D shape, and therefore its ultimate function.
So form follows sequence, which dictates function.
Precisely. And that's why abnormalities in the primary structure can be so devastating. The source material really emphasizes this. A vast majority of known genetic diseases, things like sickle cell anemia, cystic fibrosis, they stem from tiny errors in this primary sequence. Often just a single amino acid change in a chain hundreds long.
Just one wrong brick can compromise the whole building. And the source makes a distinction between polypeptide and protein.
Yeah, it's a bit arbitrary, based on size. Generally, a chain with more than 10 amino acids is called a polypeptide. If it's longer, usually more than 50 amino acids, and typically folded into a functional shape, we tend to call it a protein. But the underlying principle is identical. The sequence is paramount.
Okay, so what actually holds these amino acids together in that specific sequence? What's the glue?
The glue is an incredibly strong covalent bond called the peptide bond. This bond forms specifically between the alpha carboxyl group, a MATHM-COOH, one amino acid, and the alpha amino group, a MATHM, the next amino acid in the chain.
And water gets released, right? A dehydration reaction.
Yes, exactly. A molecule of water H2O is removed in the process. So you end up with this MATHM-CONH2 linkage between the two amino acid residues.
And these bonds, they're the real cementing material of the protein world, as the source puts it. They're strong.
Very strong, covalently. But they also have some really important characteristics regarding their geometry. They are rigid and planar.
Planar, meaning the atoms involved lie flat.
Yes, the CO group, the NH group, and the two alpha carbons they connect all lie roughly in the same plane. This is because the peptide bond has partial double bond character. Resonance between the CO and CN bonds prevents free rotation around that CN axis.
Ah, okay. So it's not like a freely rotating single bond. That rigidity must limit how the backbone can twist and turn.
Critically important. It significantly limits the possible conformations the polypeptide backbone can adopt, which makes the folding process much more constrained and predictable, contributing to the stability of the final structure.
And they usually exist in a specific configuration. Trans.
Yes, the trans configuration is strongly favored. This means the alpha carbons, and thus the R groups, of adjacent amino acids are on opposite sides of the peptide bond. This minimizes steric hindrance, or bumping, between those potentially bulky R groups. The cis form, where they're on the same side, is much rarer, usually only seen with proline.
Okay, trans is standard, and even though the bond itself is strong, parts of it are polar.
That's crucial. The carbonyl oxygen has a partial negative charge, and the amide hydrogen has a partial positive charge. This polarity within the backbone itself is absolutely essential.
Why?
Because these are the very groups that will participate in forming the hydrogen bonds that stabilize the next level of structure, the secondary structure, like alpha helices and beta sheets. The backbone itself provides the H-bond donors and acceptors.
Got it. So the peptide bond provides strength, rigidity, and the H-bonding capability for later folding. And we always read the sequence in one direction.
Yes, the convention is absolutely fixed. The sequence is always read and written, starting from the N-terminal residue, the amino acid at the end, with the free, unbonded alpha amino group. That's considered the beginning or the left side.
N for nitrogen, N for numero uno.
If that helps. And it ends at the C-terminal residue, the amino acid at the other end, with the free, unbonded alpha carboxyl group. That's the finish, or the right side.
N terminus to C terminus, left to right.
And this convention isn't just arbitrary. It actually mirrors the direction in which proteins are synthesized by the ribosome in the cell. Synthesis starts at the N-terminus and proceeds towards the C-terminus.
Okay, so we know the sequence is vital, but how did scientists actually figure out the primary structure of a protein? Especially back in the day before we could just sequence the gene. It sounds incredibly complex.
It was, and still is, a complex undertaking requiring meticulous work. The source outlines a classical three-stage approach, which always starts with getting a highly purified sample of the protein you're interested in.
Right, you need to know you're only sequencing one thing. What's stage one?
Stage one is determining the overall amino acid composition. This doesn't tell you the order, just what amino acids are present and in what relative amounts.
How do you do that?
You take your pure protein and completely break it down into its individual amino acids. This requires complete hydrolysis, breaking all the peptide bonds. Usually done with strong acid, like 6-MHCl at a high temperature for many hours.
That sounds pretty harsh.
It is, but it gets the job done. Then, you take that mixture of free amino acids and separate them, typically using ion exchange chromatography. As the different amino acids come off the column, you react them with a reagent like 9-hydrin, which makes them colored, and quantify the amount of each one.
Okay, so stage 1, know your inventory of bricks. What's stage 2?
Stage 2 is fragmentation. Most proteins are way too long to sequence in one go, even with modern methods. So you need to break the long polypeptide chain into smaller, more manageable peptide fragments.
Okay, chop it up. But how do you do that reliably?
Well, first, if your protein is made of multiple polypeptide chains, subunits, held together by non-covalent forces, you need to separate those using denaturing agents like urea or guanidine hydrochloride. And if there are any disulfide bonds holding chains together or forming loops within a chain, those need to be broken too, usually by chemical reduction in alkylation or oxidation with performic acid.
Right, get down to individual linear chains first, then chop.
Then you chop using highly specific methods. Enzymatic cleavage is very common. The enzyme trypsin is a workhorse here. It's a protease that cuts the polypeptide chain very specifically, but only on the carboxyl side of lysine, like E's, K, or arginine, R residues.
So it cuts after K or R, predictable cuts.
Highly predictable. Another enzyme, chymotrypsin, cuts after large hydrophobic residues like phenylalanine, Phe-F, tryptophan, Trep, W, and tyrosine, T or Y. Using different enzymes gives you different sets of fragments.
Are there chemical ways to cut too?
Yes. Chemical cleavage. The classic example mentioned in the source is cyanogen bromide, Atherm-Cianber. This chemical reagent is unique because it specifically cleaves the peptide bond only on the carboxyl side of methionine, Met-M residues.
After methionine. Okay, so using trypsin gives one set of fragments, CNBR gives another set. Why do you need different sets?
Because that's how you figure out the order. By generating different sets of overlapping fragments, you can piece the full sequence back together like a jigsaw puzzle. That overlap is absolutely key for the final assembly stage.
Ah, okay. Fragmentation gives you puzzle pieces. What's stage 3?
Stage 3 is the actual sequence determination of those individual peptide fragments, figuring out the amino acid order within each small piece.
How's that done?
Historically, the first major breakthrough was Sanger's reagent, which is FDNB, 1-fluoro-2,2-nitrobenzene. Sanger used this to determine the sequence of insulin Nobel Prize-winning work. FDNB reacts specifically with the free N-terminal amino group of a peptide.
So the tag's the first amino acid.
It tags the N-terminal amino acid, but the downside was, to identify which amino acid got tagged, you then had to completely hydrolyze the rest of the peptide chain with acid. So you found out the first amino acid, but you destroyed the rest of the fragment in the process. You'd have to start over with more material to find the second one. Very laborious.
Like finding the first brick, but having to demolish the wall to see it, as the outline said, not ideal.
Not ideal at all. The real revolution came with Edmond degradation, developed by Per Edmond. What's truly fascinating here is the elegance of his chemistry, using Edmond's reagent, which is phenyl isothiocyanate, PITC.
How does that work?
PITC also reacts with the N-terminal amino group, but under mild acidic conditions, it allows you to cleave off only that first amino acid as a derivative, a PTH amino acid, phenylethiodiantoine derivative, leaving the rest of the peptide chain completely intact.
Whoa, so you identify the first one and the rest of the peptide is still there.
Exactly. It allows for sequential degradation. You identify the first PTH amino acid, then you take the remaining peptide, now one residue shorter, and react it again with PITC to identify the new N-terminal amino acid. You could repeat this cycle over and over again.
Peeling off the amino acids one by one from the N-terminus. That's brilliant.
It was a game changer. This process was later automated into a machine called a sequenator. The machine performs the Edmond chemistry cycles automatically and identifies each released PTH amino acid as it comes off, usually using high-performance liquid chromatography, HPLC.
Okay, so the sequenator sequences the fragments, but how do you get the full protein sequence?
That's where those overlapping peptides come back in. You sequence all the fragments you got from cutting with trypsin, then you sequence all the fragments you got from cutting with CNBR. Because the cut sites are different, the fragments will overlap.
So a fragment from the trypsin digest might start halfway through a CNBR fragment and end halfway through the next one.
Exactly. By finding these overlaps, you can logically deduce the correct order of all the fragments, and therefore reconstruct the entire original primary sequence of the protein. It's like putting the jigsaw puzzle together using the edges of the pieces.
Okay, we've built the primary chain, the linear sequence, but a straight chain doesn't really do much, does it? The function comes from the folding.
Precisely. The linear sequence contains all the information needed for the protein to spontaneously fold into its unique and functional three-dimensional shape. This folding process happens in a hierarchy of structural levels, starting with secondary structure. This is the first level of actual folding. It refers to the regular, repeating spatial arrangements of adjacent amino acid residues in the polypeptide chain. It's about local folding patterns.
Not the whole protein shape yet, just local segments twisting or arranging themselves.
Exactly. And the entire premise of secondary structure relies on one key stabilizing force: extensive hydrogen bonding. But critically, it's hydrogen bonding between the backbone atoms, the CO and NH groups of the peptide bonds themselves, not involving the R groups at this stage.
Okay, backbone H bonds hold secondary structure together. What are the main types?
The two major, most stable types are the alpha helix and the beta pleated sheet. The beta helix is probably the most common. It's a tightly packed, coiled structure, almost always right-handed, like a standard screw thread.
Like a spiral staircase.
A bit like that, yes. It's stabilized by hydrogen bonds formed within the helix itself. Specifically, the CO group of one amino acid, let's call it residue N, forms an H bond with the NH group of the amino acid that is 4 residues further down the chain, residue N plus 4.
Okay, N to N plus 4 bonding. That sounds like a very regular pattern.
It's extremely regular. This pattern ensures that almost all the peptide bonds, CO and NH groups within the helix, except for the first few NH groups at the start and the last few CO groups at the end, are participating in this hydrogen bonding network. It maximizes stability.
And how many amino acids does it take to make one turn of the helix?
It averages 3.6 amino acid residues per turn. And another key feature is that the R groups, the side chains, project outward from the helical axis.
Sticking out from the spiral, why is that important?
It minimizes steric clashes.
Yeah.
If the R groups were pointed inward, the core would be too crowded, especially with bulky side chains. Pointing outward allows them to interact with the environment or other parts of the protein later on.
Okay. Alpha helix, right-handed coil, 3.6 residues turn, N to N plus 4 H bonds, R groups out. Are there amino acids that don't like being in an alpha helix?
Yes, definitely. We mentioned proline earlier.
The kink inducer.
Right. Because its side chain is bonded back to the backbone nitrogen, proline lacks the NH group needed to donate that crucial hydrogen bond within the helix. Plus, its rigid ring structure doesn't fit the required helical angles. So proline is often called a helix breaker. It disrupts or terminates an alpha helix.
Any others?
Also, having a long stretch of amino acids with the same charge, say, several glutamates or lysines in a row, can be disruptive due to electrostatic repulsion between the R groups. And very bulky R groups close together can sometimes cause steric hindrance.
Okay, so alpha helix is one major type. What's the other?
The other is the beta pleated sheet. Unlike the coiled helix, beta sheets are formed from polypeptide segments that are almost fully extended. Think of them as stretched out strands.
Beta strands.
Yes, beta strands. A beta sheet is composed of 2 or more of these beta strands lying side by side. The stabilization again comes from hydrogen bonds between the backbone CO and NH groups.
But if the strands are side by side, the H bonds must form between the strands, right? Not within a single strand like the helix.
Exactly. The H bonds form between adjacent beta strands. These can be strands from different polypeptide chains, interchain H bonds, or more commonly, segments of a single polypeptide chain that fold back on themselves, interchain H bonds.
Okay. And how are the strands arranged relative to each other?
They can be arranged in 2 ways. In a parallel beta sheet, the adjacent strands run in the same direction. Example, both N terminus to C terminus. In an anti-parallel beta sheet, the adjacent strands run in opposite directions.
Does that affect the structure?
Slightly. The hydrogen bonding pattern is a bit different, and anti-parallel sheets tend to be slightly more stable. The R groups in a beta sheet project alternately above and below the plane of the sheet, creating that pleated appearance.
Okay, so extended strands, H bonded side by side, parallel or anti-parallel, R groups alternating up and down. Where do you typically find beta sheets?
They are often found forming the rigid core structure in many larger globular proteins. They provide a strong, stable framework. Think of silk fiber, for instance. Its strength comes largely from extensive anti-parallel beta sheets.
All right, so we've got local coils, alpha helices, and sheets, beta sheets, forming in the secondary structure. What happens next? How does the whole chain fold up?
That brings us to the tertiary structure. This level describes the overall, complete three-dimensional arrangement of the entire polypeptide chain, including how those alpha helices and beta sheets, plus any loops or turns connecting them, fold up relative to each other in space.
So this is the final 3D shape of a single polypeptide chain, the functional shape.
Exactly. This is the level where the protein typically achieves its biological activity. It results in a compact, often roughly spherical, globular, or elongated fibrous structure.
And what drives this overall folding? We talked about polarity.
Polarity is huge here, especially for globular proteins folding in water. The formation of the hydrophobic core is arguably the single most important driving force. The polypeptide chain folds spontaneously in such a way as to bury the hydrophobic nonpolar R groups in the interior, minimizing their unfavorable contact with water.
Hiding the oily bits again.
Hiding the oily bits. Simultaneously, the hydrophilic polar and charged R groups are preferentially positioned on the exterior surface, where they can happily interact with the surrounding water molecules or other cellulose and locked into place by a whole range of interactions, primarily involving the R groups this time. We have both covalent and forces at play.
Okay, what's the covalent one? There's only one main type, right?
The only covalent bond that typically contributes to stabilizing tertiary and sometimes quaternary structure is the disulfide bond.
Formed between two cysteines.
Yes, formed by the oxidation of the sulfhydrylyl groups of two cysteine residues, creating a strong covalent link between different parts of the polypeptide chain, or sometimes between different chains. These disulfide are permanent structural staples.
And you find them more often in proteins that exist outside the cell.
Very often, yes. Proteins that are secreted from the cell, like antibodies or digestive enzymes or hormones like insulin, often rely heavily on disulfide bonds for stability, because the extracellular environment can be much harsher, more oxidizing variable pH than the inside of the cell.
Okay, disulfide bonds are the covalent glue. What about the non-covalent forces? These must be important too.
Hugely important. In fact, for most proteins, especially intracellular ones, the tertiary structure is primarily maintained by the cumulative effect of a vast number of relatively weak non-covalent interactions.
What kinds?
There are several key types. Hydrogen bonds can form between polar R groups, like serine, veterinine, asparagine, glutamine, or between polar R groups and backbone atoms, or between R groups and water. Ionic interactions, also called salt bridges, occur between positively charged basic R groups, all ice, arg, his, and negatively charged acidic R groups, asp, glue.
The positive-negative attraction.
Exactly. Then, of course, there are the hydrophobic interactions, the tendency of nonpolar R groups to cluster together away from water, which we said is a major driving force for folding itself. And finally, there are the very weak, short-range van der Waals forces, which are attractive forces that occur between any two atoms that are very close together.
So it's the sum total of all these weaker forces, H bonds, ionic bonds, hydrophobic packing, van der Waals, plus maybe some disulfide staples, that holds the whole 3D structure together.
Precisely. It's a collective effort. Lots and lots of weak interactions add up to create a stable, specific tertiary structure.
And sometimes within that overall tertiary structure, there are distinct regions, domains.
Yes, that's a very important concept, especially for larger proteins. Domains are distinct, structurally stable, and often functionally independent regions within a single polypeptide chain. A large protein might consist of two or more domains, each perhaps responsible for a different aspect of the protein's function, like binding a substrate or anchoring to a membrane. Domains often fold independently of each other.
Okay, so tertiary structure is the final shape of one polypeptide chain. But some proteins are made of more than one chain, aren't they?
Yes, many are. And that brings us to the highest level of protein organization, quaternary structure. This level only applies to proteins that are composed of two or more separate polypeptide chains, which are often called subunits.
So if a protein is just a single chain, it only has primary, secondary, and tertiary structure. But if it's made of multiple chains associating together, that association is the quaternary structure.
Exactly. Such proteins are called oligomers, or multimeric proteins. A protein is a dimer, so on. Hemoglobin, the oxygen carrier in our blood.
And what holds these separate subunits together in the final complex? Is it more peptide bonds?
The critical point. The subunits in a quaternary structure are held together exclusively by non-covalent interactions.
Only non-covalent. So the same forces that stabilize but now acting between different polypeptide chains.
Precisely the same forces: hydrogen bonds, ionic interactions, salt bridges, hydrophobic interactions, often very important at the interface between subunits, and van der Waals forces. There are generally no covalent bonds linking the subunits together.
Why is that significant? Why rely only on weaker forces to hold the whole complex together?
Because it allows for flexibility and regulation. The fact that subunits are held by weaker non-covalent bonds means their relative positions and interactions can change subtly. This is the basis for allosteric regulation, which is incredibly important for controlling metabolic pathways and cellular functions.
Allosteric meaning action at a distance.
Sort of. It means that the binding of a molecule, like a substrate, activator, or inhibitor, to one site on one subunit can trigger a conformational change, a shape shift that is transmitted through the quaternary structure to affect the activity or binding properties of the other subunits.
Like in hemoglobin, binding oxygen to one subunit makes the others bind to oxygen more easily.
Exactly. That's cooperative binding, a classic example of allostery mediated by quaternary structure changes. The relatively weak non-covalent bonds between subunits allow for this essential communication and functional integration between the different parts of the protein machine. It allows for much more sophisticated regulation than a single rigid polypeptide chain could achieve.
Okay, we've assembled the protein from primary sequence right up to potentially complex quaternary structures. Now let's think about how these finished molecules actually behave, say, in a test tube or in the cell. What are some of their key physical properties? Hashtag 4.1 physical properties and colloidal nature?
Well, the most obvious property is their sheer size. Proteins are macromolecules, meaning they are very large compared to simple molecules like water or glucose. Their molecular weight varies enormously depending on the number of amino acid residues they contain.
What's the typical range?
The average molecular weight of a single amino acid residue is about 110 daltons. So a small protein like insulin, which has 51 amino acids, has a molecular weight around 5,700 Day. A medium-sized protein like hemoglobin, with about 574 residues in its 4 chains, is around 64,450 Day. And some protein complexes involved in muscle structure or DNA replication can have molecular weights in the hundreds of thousands or even millions of daltons.
So huge variation. And because they're so large, they don't really dissolve in water like salt or sugar, do they?
That's right. Due to their immense size, proteins don't form true solutions in water. Instead, they form colloidal solutions, or colloidal dispersions. The large protein molecules remain dispersed and suspended in the water, usually due to hydration layers and surface charges, and they don't settle out under gravity.
But they're too big to pass through certain membranes.
Exactly. They typically cannot pass through semi-permeable membranes, like dialysis tubing, which have pores large enough for water and small solitudes, but too small for large protein molecules. This property is widely used in biochemistry for separating proteins from smaller molecules, a process called dialysis.
Okay, colloidal nature is important. How else do we classify proteins based on their properties? The source mentioned solubility and composition.
Yes, there are broad classifications based on chemical nature and solubility. The simplest category is simple proteins. Upon hydrolysis, these yield only amino acid residues and nothing else.
Examples.
Albumins, like serum albumin in blood, soluble in water, and globulins, like antibodies, often soluble in dilute salt solutions, but not pure water, are common examples of globular simple proteins. Fibrous simple proteins include things like keratins, insoluble structural, and collagens, insoluble structural.
Okay, simple means only amino acids. What's the next type?
Conjugated proteins. These are proteins that, upon hydrolysis, yield not only amino acids, but also some non-protein component, which is called a prosthetic group. This prosthetic group is essential for the protein's biological activity.
So it's a protein plus something else attached, like what?
Lots of possibilities.
Yeah.
Nucleoproteins have nucleic acid, DNA or RNA attached, like ribosomes or chromosomes. Glycoproteins have carbohydrate chains attached, common for cell surface receptors and secreted proteins. Lipoproteins have lipids attached, crucial for transporting fats in the blood. Chromoproteins have a colored pigment as their prosthetic group.
Like hemoglobin, its prosthetic group is the iron-containing heme group, which gives blood its red color.
Exactly. The heme group is essential for oxygen binding. Without it, the globin protein part is non-functional for oxygen transport.
Okay, simple and conjugated. Is there a third category?
The source also mentions derived proteins. This isn't really a structural class, but rather refers to products formed when native proteins are altered by heat, enzymes, or chemical agents. It includes denatured proteins or smaller peptide fragments produced by partial hydrolysis.
All right, so proteins are big, colloidal, and can be simple or conjugated. But maybe the most defining physical property, the one that really dictates how a protein behaves in solution, especially in response to pH changes, is its isoelectric pH, or PI. So what does this all mean?
The PI is absolutely fundamental. It's defined as the specific pH value at which the protein molecule exists as a zwitterion and carries zero net electrical charge.
Zero net charge, meaning the total number of positive charges on the protein exactly balances the total number of negative charges.
Precisely. Remember, proteins have many acidic R groups, asp, glue, C terminus, that can be negatively charged, and many basic R groups, mol, Is, arg, his, N terminus, that can be positively charged, mothrum, NH3, or similar. The PI is the unique pH, where the sum of all positive charges equals the sum of all negative charges.
And the value of the PI must depend heavily on the amino acid composition.
Absolutely. A protein with a high proportion of acidic amino acids, asp, glue, will have a low PI, meaning it needs a very acidic environment, low pH, to protonate enough groups to reach neutrality. Conversely, a protein rich in basic amino acids, Lys, arg, his, will have a high PI, requiring an alkaline environment, high pH, to deprotonate enough groups to become neutral.
Can you give some examples?
Sure. Pepsin, the digestive enzyme that works in the highly acidic environment of the stomach, pH 1 and 2, has a very low PI, around 1.1. Casein, the main protein in milk, has a PI of 4.6. Human serum albumin is around 4.9. Lysozyme, an enzyme found in tears and egg white that has many basic residues, has a high PI of about 11.0.
Wow. Huge range from 1.1 to 11.0. Okay, so the PI is the pH of net neutrality. Why is this so important practically?
Because at its PI, a protein exhibits several key properties. Since it has no net charge, the repulsive forces between protein molecules are minimized, and the attractive forces can dominate. Also, its interaction with water is often weakest at the PI. Therefore, at its isoelectric pH, a protein typically exhibits minimum solubility.
Least soluble when neutral.
And, consequently, it exhibits